one more minute. Welcome everyone. It's a real pleasure and honor to begin this first symposium of Algorithmiq. This is a symposium that brings together different fields, and in particular, quantum physics, network medicine, and network sciences. I am Sabrina Maniscalco. I'm a professor of quantum computing and logic at the University of Helsinki, and I am also the CEO and co-founder of Algorithmiq. And we decided that it would be very nice to have an informal, but also full of discussions type of meeting in which we would like to try to bring together disciplines that we believe are different but complement each other. And so ideally we would like to have a new take on several of the topics that we will be discussing. So I'm very glad and honored to have here very well-known speakers that are extremely famous, outstanding figures in all of these fields. And I will begin just by briefly discussing the program, and in fact we will have a change in program, and in particular one of the speakers will not be able to attend, so unfortunately Professor Martin Plenio that had to come here and talk about quantum biology was unable to come because of health reasons, and therefore we will be discussing with the other three speakers. There will be, I think, some touch points with quantum biology as well, and I know that this is just the beginning of a future interaction and exchanges. So I know that Martin will be able and will be very interesting, is very interesting to continue our discussions in the future, because we hope that this is just the beginning of collaborations and of interactions and of potentially what can be a new emerging discipline. So I will say a few words first about Algorithmiq. Algorithmiq is our startup focusing on quantum software and algorithms for life sciences in particular. So we are interested in developing software and algorithmic enhancement using quantum computers in what is the specific field of the life sciences, beginning from quantum chemistry, but as we will see later on, going all the way to drug development and discovery at large. So we have a plan that is very ambitious, but that it is, we believe, very topical, because during these recent years, advances in quantum computers have been remarkable, and we are very close to a moment in which we will be able to use these devices for something that is very impactful in several fields, including the life sciences. So the program will begin with Professor Loscalzo from Harvard Medical School. We will continue with Professor Mariangeles Serrano, and then Professor Tony Acin. And in the end, we will have the Chief Scientific Officer of Algorithmiq, Guillermo Garcia-Perez, who will talk about what we think is a way or is a possibility of combining these three fields in what will be, we believe, an interesting new avenue for both network medicine and quantum algorithms. So I think that also to add to the specific program, I would like to mention that it would be really desirable for us to have discussions, several discussions. So we structure the program in a way that we will have the standard 40 minutes presentation, but then at least 10 minutes for discussions with the audience. And we will be able also to accept questions from the people who are following online via, I think Boris, Boris will be reading the questions and then we will be answering. So there is a chat that you can access for ask questions online as well. And then for the people who are here in the audience, I would like also to encourage them during because we don't have official coffee breaks, but during the 10 minutes breaks to just maybe move and go and get some water or some coffee. So the 10 minutes of question and answer can be also an occasion to move, not just to stay sit all the time. So I encourage you also to be open to move in the room during the question and answer. Okay, this is a brief introduction, and I think that at this point we are ready to begin with the first speaker that I will introduce. Professor Joseph Loscalzo is a Hersey Professor of the Theory and Practice of Medicine at Harvard Medical School. He's the chairman of the Department of Medicine and physician-in-chief at Brigham and Women's Hospital. Professor Loscalzo is recognized as an outstanding cardiovascular scientist, clinician, and teacher. He has received many awards, including the Clinician-Scientist Award, the Distinguished Scientist Award, the Research Achievement Award, the Paul Dudley White Award, and the American Heart Association, from the American Heart Association. And what is also remarkable is that together with Professor Barabasi, he is really one of the fathers of the emerging field of network medicine, which we have been following, and we find really a discipline that will be groundbreaking, that is beginning really to open new possibilities and directions in the field of medicine and systems biology. So, Professor Loscalzo, we are very honored to have you here, and I will now pass you the microphone for your presentation.
Well, thank you very much for that kind introduction, Sabrina. I'm really delighted to be here today to talk with you a little bit about network medicine and how we're beginning to use and develop this field to address causes of human disease, especially complex phenotypes, as well as therapeutic strategies. Here are my disclosures. So, as everyone in this audience recognizes, but many biologists do not, reductionism really does provide limited insight in understanding biological complexity and complex disease phenotypes of the sort we deal with as adult physicians. And it has, as you know, a rich history, including that of Democritus, the Greek philosopher, the atomist, as well as, of course, Descartes, who set the stage for the scientific approach that we've used for the past 250 years or so. And basically, reductionism forces us to seek ever more refined associations that ultimately can define causation of a phenomenon. But its limitations have been increasingly appreciated, especially in this era of omics and big data in biology. Weighted genome-wide association studies, polygenic risk scores, etc., really do little to address the problem of complexity in biological systems. Now, there are two major problems with the reductionist approach to disease. The first is it reflects the linear narrative notion, and by its very nature, oversimplification. We communicate logically in a linear way, and it really reflects, that linear narrative reflects our efforts to carve a hyper-focused, rational path through a morass of information. The second major problem, of course, is the age-old correlation-causation challenge, our ingrained desire to explain the inexplicable simply. An eclipse leads to death and destruction, as one simple example. Now, there's a long arc of reductionism in biomedicine that dates back really to Morgagni and then to Osler. And in a piece I wrote with my colleague Jeremy Green, who's an historian of medicine at Johns Hopkins, we used as an example of this arc the approach to the patient with tuberculosis. Beginning in the 18th century, tuberculosis wasn't really understood as we know it now. It was referred to as phthisis. It reflected, in fact, chronic consumption and ultimately death. Into the 19th century, with early forms of microscopy, lesions of organs and tissues were recognized that were unique. The infection-causing granulomas, which are the hallmark of the disease histologically. In the latter part of the 19th century, lesions of cells and microbes were recognized as important in the mechanism of disease, and the identification of Mycobacterium tuberculosis by Koch was established. And then ultimately, in the later part of the 20th century, we know how to treat the disease, we can cure the disease. Importantly, even before antituberculous therapy developed, the disease was on the wane worldwide, which really reflects an ecological transition that we rarely take into consideration as we think about relationships between hosts and microbes. Now, I should say that that Morgagni and then Osler really established the one syndrome, one organ, one disease notion that we follow in medicine. And of course, it is based on clinical pathologic correlation at autopsy. The problem is, of course, that our challenge today is to move back in time, both from the perspective of disease within a patient, as well as historically, and begin to reconstruct the entire system to understand it in a much more mechanistically rich and important way. But even in modern genomics, we continue to use the linear narrative to express our understanding of disease. Here's a Manhattan plot. For those of you who don't look at these, this is really on the x-axis. You can see the chromosomes in a typical cell, and the green dots reflect associations at a particular site or locus in a chromosome with a specific disease. Now, these are purely statistical associations. They have given insight into specific mechanisms, but they, as you might imagine, don't necessarily give us insight into interactions between loci, because the statistical power of this kind of analysis is limited, especially for less common diseases. So therefore, it's important to really think about genomic context as we think about even the most straightforward diseases. And I'll set the stage by picking a conventional Mendelian disorder. And in this case, Mendelian refers to a disease for which there's believed to be a single cause, single genetic cause, and which is 100% penetrant, which is to say that everyone who has the mutation has the disease. If that were the case, then patients with sickle cell anemia would all have the same phenotype. And clinicians recognize that is not at all the case, that some people have severe hemolysis or lysis of red cells. Others have stroke syndromes. Others have acute chest pain syndromes. And there are a host of other subtle changes in the way diseases present, which really reflect the genetic context or genomic context within which a major genetic driver operates. So that point was really driven home in a paper by Chen and Steve Friend's group now in 2016, in which they analyzed over almost 600,000 genomes for diseases that are very specific to sorry. Is that better? So in this analysis by Chen and friend, they screened 874 genes found in 13 adults who bore 8 mutations for severe Mendelian conditions, meaning that if they bore these mutations, it was expected that they would have a severe clinical syndrome. And none of these individuals had clinical disease. So they concluded that these, quote, individuals are buffering the effects of rare, highly penetrant, deleterious mutations, unquote. Well, that's obvious. The question is how. So over the last 15 years or so, and when I say we, in this case, I'm referring to a group of us, beginning with work that Barabasi and I did together when he took a sabbatical in Boston before he moved from Notre Dame and began to get involved in biological networks, and Ed Silverman, who's the head of our network medicine division, who's a pulmonary geneticist. The three of us have been working to establish this new field of network medicine, and we do so based on the following principles: that most biological systems are complex systems that form molecular networks, and I'll describe specifically what I mean by molecular networks in a moment, and new quantitative approaches can be used to analyze these networks that have become available over the last 15 to 20 years. So this new field applies these approaches to disease pathobiology and treatment. So the first question we can ask is, can molecular interaction networks define genomic context of the sort that was apparent in the work of Chen and friend, and give unique insight, therefore, into disease pathogenesis? And in a concept paper written now over a decade ago, Laszlo and I proposed that, in fact, if one looks at the complex molecular interaction network, the interactome, which in this case is the protein-protein interaction network, that within that network there are modules or subnetworks which are specific to a particular disease. And that hypothesis then turned out to be correct, as I'll show you in a moment. The basis for that hypothesis are these 4 other hypotheses and organizing principles. First, the local hypothesis that proteins involved in the same disease tend to interact. Then the disease module hypothesis that proteins involved in the same disease tend to cluster in these connected subnetworks. The functional coherence hypothesis that proteins in a disease module are often involved in the same biological process or mechanism. And the shared components hypothesis that related diseases are located in the same interactome neighborhood from which unrelated diseases are excluded or separated. So in a paper in 2015, led by Jörg Menke, who was a postdoc at the time, this principle, this notion, this hypothesis that in fact there are submodules specific to particular diseases in the global protein-protein interaction network was proven to be true. And here we curated all experimentally validated protein-protein interactions in the human cell to build this network. It began with the binary PPI that was first developed by Mark Vidal using a yeast-2 hybrid screening method. But it included other curated data sets in the literature, as well as unbiased data sets. And as you can see, the sort of fuzzy background reflects the PPI that we had available at the time, that included over 13,000 nodes or proteins linked by over 140,000 edges or physical interactions. Now, those in the audience with a background in biology will remember when you took your first biochemistry course, the professor pointed out that the reason proteins are as big as they are was because the very precise mechanisms required for catalysis or for binding required this infrastructure to stabilize those sites optimally. But that, of course, is nonsense, that you can build an enzyme with far fewer amino acids or other chemical structures, as well as ligands for binding proteins. The reason proteins are as big as they are is because, as recognized by this ratio here of about a little more than 10 to 1. That is, each protein in a cell interacts with 10 other proteins. And those interactions then lead to an extraordinary complexity and richness of the microenvironment of the cell that governs complex behaviors. So how do we derive a disease module? Here's an example of one such derivation by my colleague Rusheng Wang. We first identify a disease of interest. In this particular case, it was cerebrovascular disease or stroke. Then we ascertain disease network components from the literature or our own work. This was all literature-based. These are so-called seed proteins, which are shown in blue. We map the seed proteins to the interactome, and then we identify those connector proteins shown in gray, which link seed proteins by not more than one edge. We then define the resulting disease module within the interactome on a statistical basis. We show that it's clustered and distinctly compact compared to an equal number of proteins randomly chosen from the interaction network. And when we applied this principle in the Menke paper to 299 different diseases, we were able to identify discrete disease modules in about 85% of them. Part of the reason we didn't identify more is likely a consequence of the fact that the interactome is incomplete, and according to our best estimates currently probably contains about 30% of the possible protein-protein interactions in a cell. So we and others have used this principle, this notion of disease module discreteness, and the fact that these connector proteins fill in the missing links for pathways and drug targets and mechanistic drivers to analyze and identify novel mechanisms for well-established diseases. And I won't go through these in detail. You can see the references here on the slide if you're interested, but they range widely from preeclampsia in pregnant women, a form of high blood pressure that can be lethal, to type 2 diabetes, to regulatory features of asthma, to calcification of the aortic valve, a particularly problematic illness in older folks. And I'm going to touch a little bit on PAH, which is pulmonary arterial hypertension and vascular fibrosis, and HCM, which is hypertrophic cardiomyopathy, in a little bit. So let's just return for 2 slides to this question: how do genetic variants affect these protein-protein interactions? And can we gather any more insight, knowing the protein-protein interaction network, insight about mutations that might drive disease phenotype? Well, a work which Fei-Sheng Cheng did when he was a postdoc with us, published now last year, he asked the following question. He asked whether or not mutations that are believed to be associated with a particular disease, in this case germline mutations, not cancers, that mutations associated with germline diseases, are they more or less likely to be located at the interface between 2 interacting proteins? And as you could see in the yellow stars here crosses on the slide, the answer was resoundingly positive for those mutations being more likely to occur at the interface binding site rather than the exofacial sites. And as you can see in the graph at the bottom, there was about a 4 to 6-fold increase in the likelihood that those mutations were interfacial mutations. Obviously, to perform this analysis, we needed to know the 3D structure of these proteins and their structures in complex, either directly measured by X-ray crystallography or by NMR, or imputed from other portions of proteins or orthologous proteins. He performed the same analysis with somatic mutations, in this case malignancies, of which there were 33 cancer types that he analyzed with the same principles in mind. And of those 33, I believe there were 31 that showed the same highly statistically significant difference. Now, the fold difference was about 6 to 8-fold. That is, mutations were more likely to affect PPIs than non-interfacial sites in these malignancies. So that really highlights the importance of disease drivers, genetic disease drivers, in their influence on the protein-protein interaction network and its consequences for disease mechanism. So the next question we can ask is, can PPI networks facilitate personalized genomic medicine? This has been the promise, of course, in genetics and genomics for the last 20 years and really not very well realized, largely because people continue to focus on genome-wide association studies and derivative reductionist approaches that still are not very informative. Well, to give you an example which will answer this question, I want to turn to this disease, hypertrophic cardiomyopathy, which is actually the most common genetic cause of heart failure. there are mutations that have been identified in 11 sarcomeric proteins. These are the proteins that sit in the portion of the contracting cell that contains the contractile proteins, actin, myosin, tropomyosin, troponin. There are over 1,400 variants that have been identified in these proteins, in these genes, and these variants are heritable. But there's highly variable penetrance of this disease. In fact, so much so that only about a third of patients have the mutations. The other 2/3 do not. So the phenotype is identified not at autopsy, ideally, but by echocardiography, where a very thick left ventricle, which is highlighted here, can be identified. There are many causes for very thick left ventricular walls, but HCM is the predominant genetic cause. But because of the fact that 2/3 of patients don't have sarcomeric mutations, we, and in this case, we as Brad Marin and Rusheng Wang in our group, decided to analyze the data in a different way, in a network-centric way. And what are the data that we used? Well, the first is that we wanted to obtain information about expressed genes in the involved tissue. As it turns out, that was relatively easy to accomplish because patients with this disease, if they have frequent loss of consciousness or chest pain or shortness of breath when they exert themselves, are treated with medications first, but if that fails, they at times have to undergo surgery where a portion of that very thick septum, the wall separating the left and right ventricles, is excised surgically to improve blood outflow. And that excised tissue was then used as a source for the transcriptome from these patients. And the controls were explanted hearts that were about to be used for transplantation, but weren't used because of problems with the recipient. So in this case, Rusheng performed a perturbation correlation matrix analysis and did so with all of the pairs of proteins that were analyzed throughout these differentially expressed gene sets. And those that had the greatest, there was a predefined statistical threshold that was predicated on the effect on the integrity of the interaction network and the disease module. Those that exceeded that specific threshold, statistical threshold, were then mapped to the interactome to create individual interactomes for this disease module, which I call reticulotypes or reticulomes after the Latin for network. And here you can see in the 18 patients we studied, even from this altitude, that there are very significant differences in the shape, the density, the linkage of each of these networks. And that we found fascinating because we had a convergent phenotype that for all the world at least grossly looked very similar, but yet there were very different disease drivers that appeared to be associated with those phenotypes. And we in this paper, we subcategorized those drivers based on pathways that were principally activated. One of which, for example, is fibrosis, or the deposition of scar tissue outside the cell in the heart. People really had not recognized fibrosis as an important determinant of this disease phenotype. Although the original papers suggested it was important, no one had really explored it in the modern era, that is about 50 years after the disease was first recognized. And what Brad and colleagues were able to show was that if about 20% of these patients had a reticulotype in which the pathways that are responsible for fibrosis were activated and major drivers of the disease phenotype. And in fact, he was able to identify a novel primary transcription factor that appears to be a key hierarchical driver of fibrosis called JAK2, which is recognized in the cancer field as a target for certain cancers, but hadn't been viewed as important outside cancers. So we're now using that as a target for drug development for HCM. So in our construct, network-based precision medicine moves from the genotype through the reticulotype to a detailed and nuanced phenotype. I don't have time today to go through ways by which we can explore phenotypes in much more, in fact, network-based, with much more network-based precision. And through those correlations begin to define precise targets for therapies for these complex diseases. So now in the next 15 minutes or so, let me talk a little bit about PPI networks and their use in facilitating drug target identification and guide drug repurposing as one strategy for identifying novel therapies among an existing pool of approved drugs. Now, just as with Morgagni and Osler, we have one person to thank for our reductionist view of pharmacology, and he, of course, is Ehrlich, who was an organic chemist, a dye chemist, and really the first medicinal chemist or chemical biologist, the first person to perform a low throughput assay, grabbing drugs off his shelf or chemicals off his shelf to, in the first case, test them against syphilis. And the insight that he had, which was quite a stunning insight at the time, was as a dye chemist, he noticed that tissues stained differentially with different dyes. And he argued that there might be a sort of side chain, the term receptor wasn't yet in our vocabulary, that interacted with those dyes that was tissue-specific. And these tissue-specific receptors were then an outgrowth of this side chain theory. And because of his success with the identification of an arsenical as a treatment for syphilis, it became common practice to find a magic bullet, as he put it, for each disease. And that, of course, has led to what we know as the modern era of drug development. The regulatory agencies like the FDA and EMA require that a drug company identify the target before a test compound is used in a clinical trial for which the company seeks approval. But as you can see in this slide taken courteously provided by Harold Schmidt, there is a clear decay in the yield of new drugs, and that appears to will continue, at least from extrapolation, over the next 10 years. And this is really a consequence of many factors. If you talk to people in the pharmaceutical industry, they'll say that the regulatory requirements are fairly strict, and that limits their ability to achieve successful approvals. That the easy targets have all been identified and are drugged adequately. Well, those are true, but the major reason is that drug target identification is performed in a relative vacuum, and certainly never in the context of a disease module until recently. And I think a consequence of that is shown in this slide from Schork, in which he asked the question, how many patients, shown in green, do the top 10 grossing drugs benefit? This was as of 2015, I think it was, yeah. And how many, shown in red, do these drugs not benefit? And here, and this is for FDA-approved drugs, here you can see that that for every individual these drugs help, they fail to help between 3 and 24 individuals, and yet they're approved for use. So most drugs that we use as adult clinicians for common complex diseases fail to help the great majority of patients. They reach their testing in clinical trials reaches a statistical threshold that's required for approval, but that's pretty much it. And so long as they're not terrible toxicities, despite their extraordinary expense, they are approved for use and applied broadly. We have to be able to do better than this, right? So here's another principle that you may not be aware of, that drugs are dirty, which is to say that even though they're designed for treatment of a single target, which is the only target that's actually studied in the preclinical phase of the development, that according to this study by Chartier, in which they asked the question, how often does the binding site for a drug, how often is it recapitulated in the entire human proteome, just from sequence analysis? And they found that that the median was 32.4 targets per drug. So it's an enormous number of off targets. So all of you would say, well, that explains why there are these side effects that are unpredicted. Well, that's true, but I think it also offers an extraordinary possibility. There may be other targets that one could use or could use the drug for that haven't been realized previously. So there should be a great opportunity for drug repurposing here. And I'll give you some examples of that point next. So there's sort of two principles here. The one is the notion that drug targets need to be explored not just in vitro in a binding assay or an enzyme inhibition assay, but really within the pathway context, all the pathways within which they're involved. And there are many, as you know, from what I've shown you in the last few slides about PPIs. And the second principle shown on the right, that This is the proximity hypothesis, an obvious one to network folks in the audience, that if you have a disease module of interest, shown in light blue here, and drug targets that are near it but not within it, such as T2, for an entirely different disease, the question is, would it be feasible to use a drug that's designed against this target for a different disease as a treatment for this disease? Yeah. So in the first test of this hypothesis, this first effort to address this question, we worked with Sebastian Schneeweiss, who runs our pharmacoepidemiology and pharmacogenetics division within my department, and with our both experimentalists and computationalists within my lab. And the question that we asked Sebastian to address first was this. We're going to give him a disease of interest. In this case, it was coronary artery disease, heart attacks. And we wanted him to explore the incidence of heart attacks in patients who were treated with drugs, pairs of drugs shown here in the box, for diseases unrelated to heart disease, to determine whether there was a difference in the incidence over the course of, I think it was out to 18 months, of the initiation of those treatments. And these pairs of drugs were chosen with a very specific principle in mind. The targets of one of each pair had to be either within or near the coronary heart disease module, shown schematically in this big red circle, whereas the targets for the comparator drug needed to be outside, remote from the coronary disease module. So he had access to two large administrative databases that had over 200 million people in them. And these databases can give information like the onset of use of a drug, its continued use by how many times prescriptions were refilled, and the incidence of new diseases or clinical events that occurred after the drugs were started. And with that kind of analysis, and I apologize for how dense this slide is, what he was able to show, and I'm just going to focus on carbamazepine and levetiracetam, which are drugs that are used to treat seizure disorders or epilepsy, and hydroxychloroquine and leflunomide, which are used to treat rheumatologic autoimmune diseases like lupus or rheumatoid arthritis. And what he was able to show was that carbamazepine actually increased the rate of heart attacks compared to levetiracetam by about 1.6-fold, whereas hydroxychloroquine reduced the incidence of heart attacks by about 60% compared to leflunomide. So we went back to the experimental lab, and Diane Handy, in the case of hydroxychloroquine, explored its ability to suppress mechanism that's responsible for the early phases of atherosclerosis, namely activation of inflammatory markers on the endothelial cell. And what she was able to show was that these two markers, vascular cell adhesion molecule 1 and interleukin-1 beta in response to TNF-alpha, could be suppressed by hydroxychloroquine, and the protective protein endothelial nitric oxide synthase, which was suppressed by TNF-alpha, could be restored somewhat by hydroxychloroquine. So this is I think a pretty interesting way to go about drug repurposing. It involves an understanding of the network disease module concept. It involves access to very large data sets, so you have sufficient sufficient power within each cell to compare drugs. And it gives useful information that is now moving forward. For example, there's an ongoing clinical trial of hydroxychloroquine for the prevention of atherosclerotic events of heart attacks in patients without rheumatoid arthritis or lupus as a result of this and other work. Another example of repurposing is shown in this slide, and here the module is not a module for a disease per se, but a module for an intermediate phenotype, which is calcification of the blood vessel. And vascular calcifications are very important concomitant of the development of vascular disease, especially in patients with renal failure on dialysis, for example. In this case, Jun-Sup Sung and Jane Leopold identified 3 drugs, 2 of which are inhibitors of mTOR and are used as immunosuppressants, and one of which is used as a treatment for myeloma, that significantly reduced, as shown in this inset, in this in vitro assay, calcification of vascular smooth muscle cells compared to the control. So these, this also, it's not to say that we would use these drugs directly because they have other effects on the immune system, for example, but derivatives of these drugs that are chemically designed to optimize specific effects to suppress calcification would be a natural outgrowth of this kind of analysis. And more recently, working with a broader group I'll mention in a moment, we use the same principles to redesign drugs, to repurpose drugs for COVID-19, for SARS-CoV-2. Now here we have the added complexity of yet another interactome. That's the interactome within the virus. And what we did was we began by asking the question, are the human protein targets for the viral proteins, are they contained within a clustered module or subnetwork in the interactome? And from the work of Gordon, published in 2020, there were 332 human proteins that interacted with 26 of the 27 SARS-CoV-2 proteins in a yeast-2 hybrid screen. And 208 of these form a large connected component, which reflects what we call here the COVIDome. And using the same principles, repurposing candidates must target proteins in the network vicinity of this disease module. That was our hypothesis. So we used three analytical strategies here to prioritize the drugs that we were going to test. And that one included the proximity strategy that we've gone over already. Another is a diffusion strategy based on a random walker that's applied to the network, to the disease module. And the third is a network-based AI strategy for which we have Marinka Zitnik to thank, who is in Zach Kohani's biomedical informatics department, came from Stanford, where she's worked on network-based AI methodologies. And these three methodologies really gave us complementary information, as shown by the Kendall-Tau rank score here. And that's true for each of the variants. There, as you can see, 3, 5, and 4 different pipelines for these three methods. Each of the variants also gave somewhat unique information. So that complementarity led us to develop an integrated scoring system for drugs that we wanted to test in a real high-throughput screen using live virus. And in that particular case, we collaborated with Rob Davey, who runs the National Emerging Infectious Diseases Laboratory in Boston, which is the only lab that has access to live infective SARS-CoV-2 and its variants. So using this approach and testing a range of about 8 different concentrations over 3 or so log orders for over 6,000 drugs, we were able to come up with novel treatments. And without going into great detail, when we gave Rob our list of the top 40 candidates, he actually tested all 6,700 compounds in his assay, but we were highly predictive of efficacy. And I'll show you what the numbers are in our predictive accuracy, if you will. So here there are 4 drugs that we tested compared to control. The red color indicates live virus that's replicating in these cells. These cells are human intestinal epithelial cells. Remember, the virus infects intestinal epithelial cells and bronchopulmonary epithelial cells. Remdesivir was a positive control, which suppressed viral replication. The blue stain reflects living host cells, and in some cases, in these assays, we at the same time measured both virucidal activity, shown in red, as well as cytotoxic activity, shown in blue, of the host cells to get the best toxic therapeutic index. A couple of interesting compounds fell out of this analysis: prosolaridin, which is a digoxin, a cardiac glycoside derivative, and obatoclax, which is a cancer drug that's used to treat a certain kind of lymphoma. And we tested obatoclax, it's a BCL2 inhibitor, in mice that overexpressed the human receptor, ACE2, in their pulmonary epithelial cells. And we're able to show that both in female and male mice there was significant suppression of viral replication. And in data I don't have here in the slide but is in the paper, there is also a significant survival advantage. In fact, all the animals treated with obatoclax survived whereas about 85% of those treated with control died within 6 weeks. So that's yet another example of repurposing, and it's giving us insight about mechanism as well as novel drug targets and novel drug therapies that hadn't previously been recognized. So these examples, I think, really beg the question of what are we missing? How can we gather a large enough data set that would give us the chance to repurpose, to identify targets that are located in the interactome in other disease modules that for which those drugs might be useful. And to begin to address that issue, I've been working with Felice Lightstone, who's a structural biologist at the Lawrence Livermore Laboratory in California. And they use their Sierra system to do what most drug companies take a long time to do for a single target. So if you're AstraZeneca or Merck, and you have a target of interest in a 5 million compound library, and you want to screen one against the other in silico, it can take 3 to 6 months. And then you test the top hits in a high throughput assay. But with the Sierra system, which is near exascale, its latest version is, we can in fact do such an analysis with, in this case, a 2 million compound library and almost 29,000 models, three-dimensional models, tertiary structural models, for 14,000 human proteins, and have done so now over the course of about 14 months. So that will give us a large matrix of drug target associations, unbiased, which we'll make publicly available. People can explore that matrix for potential novel therapies for their favorite disease. So going back to how the network strategy can help improve the identification of drugs, I'll just give you a few statistics here. That the standard brute force screening of about 12,000 compounds, which Riva used in his Nature paper, yielded a 0.8% success rate in the high throughput screen. But drop down to the next bullet point here. We improved upon that by a factor of 10, which increased to 28% with pharmacologic curation. I was the pharmacologic curator because what I could provide as information about drugs that might be toxic, which would, for reasons that I as a clinician knew, a pharmacologist. So we eliminated those with curation. And in fact, with a smaller data set from Bruno Botta from Sapienza, we've actually improved that to 36%, using a combination of binding energy, pseudo-binding energy assessment from Felice Lightstone's analysis and network structural features in this analysis. So I think I'm getting the hook. Am I at my time? Do you know? Do I have, sorry, how close am I to time? Stop. 5 more minutes, okay? Thanks. What are the caveats for this approach? That first, we can't tell whether a computationally selected drug will improve or worsen the disease. We need to do in vitro testing to sort that out. That high throughput assays of drugs that were identified computationally can lead to adverse effects as a consequence of interactions with other targets. And that while repurposing approved drugs is assumed to be safe, new toxicities may emerge when treating a disease for which the drug was not originally intended or designed, drug by disease interactions, and that requires analysis. So given the time limitations, let me just go to my last slide, 3 last slides. So this is a summary of what I've tried to show you, that network medicine and precision therapeutics really reflect, I think, 2 ideal strategies for utilizing protein interaction networks in our improving our understanding of disease and of personalizing therapies. And we begin with deep phenotyping, common endophenotyping, or mechanisms that are common to multiple diseases, and map those to the interactome or to multiple networks that I didn't have time to talk about that interact and improve resilience in biological systems. We reticulotype individual patients or subgroups of patients, and we initiate phenotype-specific network-informed drug application. Therapy is also guided by network-based biosignatures, which is one of the things the company I co-founded, Cipher, actually focuses on. So I want to acknowledge the folks with whom we've collaborated on the left, whose names I mentioned throughout the talk, as well as those who perform the work on the right. And I'll just leave you with this slide that we could talk about in the discussion, which are unresolved issues that require computational solutions, some of which may be ideally suited for quantum computational strategies. Those include modeling PPI interfaces for proteins whose structures are not known, post-translational modification of network proteins and their functional consequences, both because these modifications are in part deterministic, such as those that involve enzymatic targeting like phosphorylation, and in part stochastic, such as oxidation of side chains of certain amino acids. Network dynamics, that is molecular information flow across disease modules and how that can be modified. Network evolution over time, disease network evolution over time, and network aging. And there are a host of others here that you can see that I'm happy to discuss, and I can leave this up for our discussion if you like. So thank you for your attention.
Thank you very much for the super interesting and fascinating talk. I think we have now time for questions. I have some, but maybe first we will take some from the audience and then from the online listener, I don't know, online attendees. So who wants to begin? Yes, Eric. Yes, Matteo will give you the microphone.
Hi, I'm Eric Aurel. I'm visiting from Stockholm, from the KTH, the Royal Institute of Technology. Thank you for a really fascinating talk.
Thank you.
A really enormous amount of questions one would like to ask. I'll just take one general question, so give the time to others also. I was involved for quite some time in the effort to take protein sequences and turn them into protein structures. I contributed one of the earlier methods, of course now very much overtaken by the development of AlphaFold, which practically solved the in silico protein structure prediction problem. And in that context, it was really important that we looked at more, let's say, advanced models than correlations between the sequences. And that's the phase when I was involved. Of course, AlphaFold has even more complicated models, which are not even entirely understandable in short terms because they are built on AI methods. But nevertheless, the idea is the same. But and I heard several talks by your colleague Barabasi on this topic, so I could identify some in your talk also. But you seem to go in the, let's say, in the network science type, where you look at this protein, this node, this identity is related or is co-varying or something with that one. And in this way, you select out what, the network and the components of the networks. And that was not enough to solve the protein structure problem. We needed to go further into mechanisms to actually finally then crack that problem as a community. So I would like to hear your thoughts on the need to find biologically and also biophysically motivated mechanisms behind the networks, and that those mechanisms, when we find them, are the ones that can be really useful to give biological and medical insight. Thank you.
Right. Well, thank you for that question. I'll answer it by touching on one of the points down here where I state need for additional data. So when we build these networks, they're static, right, these interaction networks. And we assume that two proteins that have been shown to interact in a yeast-2 hybrid screen are always going to be complex to each other. Well, that's absurd, right? Because the intracellular concentrations of those proteins will determine based on their KD. And their KD in situ, not the KD that you measure in a cell-free system, will determine whether or not those complexes form. We don't have any information on either of those 2 points. We don't know the true intercellular concentration of most proteins. We don't know the true KDs, or KAs, depending upon your preference, for those protein associations in situ. And there are a couple of interesting papers using single-cell analysis and single-molecule analysis to show that the association constants for two proteins, or for a ligand in a protein, can be off by 2 orders of magnitude when measured intracellularly, in part because of diffusion constraints in a very densely rich protein medium, as you're aware. So we first have to know the answers to that question, then we can begin to build truly, we can begin to identify the different instantiations of a network as a function of the distribution of associated proteins versus dissociated proteins, with perturbation as well as in the steady state. Having that information would then help reduce the dimensionality of that network to those features which are most important for pathways that might govern a phenotype. So I'm not answering your question as much as outlining a strategy to begin to answer the question that I think is what we as a biological community, especially with high-powered computational strategies, should be focusing on, at least in my view.
Very good. New questions, more questions? No,
Please.
is there? Yes, there is one more maybe. Yes, go ahead. We will get back to you for sure, Eric.
Hi, I'm Jari Saramäki, a network scientist at Aalto University, also Laszlo's collaborator. So I was interested in whether you are familiar with any works that would combine the ideas of network medicine with those of immunology, because in immunology there is a ton of things like the diversity of different B-cell, T-cell receptors that I feel could have touching points with this work.
Yes, and in the interest of time, I didn't show this slide. So this is a paper we just published. This is work that we performed with Mikhail Benson at Karolinska, soon to be at Karolinska, I should say. And it's a study of seasonal allergic rhinitis, which is an immunologic disease. It was published in Genome Medicine just, I think, last month. And the goal here was to do the following, to take peripheral blood mononuclear cells, lymphocytes, immunocytes, and expose them, in this case, to an allergen, birch pollen, and then to, over time, sample the subsets of those immunocytes and their single-cell RNA-seq data sets. So now you've got this incredibly rich, which I've oversimplified here in this slide, set of single-cell RNA-seq and multicellular network models. The goal of it was to try to identify upstream regulators of the immune response, just as you're suggesting, to tailor therapies. And the hypothesis was that we would find subsets, much like we did in HCM. There would be some patients who would best respond to an IL-6 inhibitor and others respond to an antihistamine. In fact, we found none of that. Every patient was really quite different in terms of the upstream regulator. Now, there wasn't a huge number of patients. Say there were, I can't remember, I think it was 18 or 20. But there was no sort of clear lead in terms of the hierarchy of transcription factors that govern the immune response. So you're absolutely right. I mean, this is another computational nightmare, not for quantum computationalists, but for us, as simple-minded people, a computational nightmare. Because if you imagine that when I showed you those data from that heart tissue, we didn't distinguish among the cell types from which we got the transcriptome, right? So there are at least 10 different cell types in that heart tissue. How can we reduce this dimensionality, both in terms of cell types and RNA drivers, to something that's meaningful? And mind you, the thing about which I worry most and don't have a good approach to yet is that many of these phenotypes could be driven by a very small subpopulation of cells and a very small subpopulation of proteins that might just give you a little bit of signal above noise. And there's no way to know, just from correlation, what's most important there. So that's another really critical, very critical issue.
Yes, I do have one question, maybe, but it's more of a general question. That is, every time there are new emerging fields, of course you have to face some challenges with what is the conventional way of thinking, in this case in medicine and so on. And my question for you was, as you were one of the person starting a new field, which were the biggest, strongest challenges you faced in the community? And what is your strategy somehow to dialogue and to address these challenges?
Yeah. So first of all, remember that this is a very interdisciplinary community. So I have to talk different languages to different people. So you can imagine when I give this talk to a bunch of physicians or a bunch of biologists, the truth is there, but it's shaped a little differently so that people can understand it. And similarly, when I speak about diseases to folks like you, I used a very different kind of language structure than I would if I were discussing it with biologists or physicians. So first thing is you really need to learn how to communicate to to all members of the interdisciplinary community to have them understand optimally what each other knows. Second is that the resistance, interestingly, I got initially was mostly from established investigators. You know, I was proudest when I first gave these talks when a young graduate student would come up to me and say, "This is great. I'm going to change the focus of my thesis. I really thank you for that insight." And then the guys my age would say, "Well, you know, look, I've been successful as a reductionist for the last 40 years. I'm not going to change now. I don't care what you say." So there's that sort of intransigence from successful people. The other point is that there's so much that these disciplines can learn from each other. I remember when I first started working with Laszlo, he said, "You know, this biology is complicated." I said, "Yeah." I said, "You know, it's not rocket science. It's much harder." We have to think about how to think through this. And there are those kinds of insights, sort of biases that we have about other disciplines that we don't know very much about, that I think limit the ability for differently trained people to communicate properly and share challenges and opportunities well. So it's basically a career-long struggle, for sure.
I can imagine. I would like to take one question maybe from the, if there are from the followers online. You can read it. Okay. Thank you, Boris.
So this had to do with the slide which was about the drug repurposing.
Yes.
And the question was, can we use ALD to improve the properties? I'm not exactly sure properties of what.
AI to improve properties?
ALD.
I'm not sure what that is. Yeah.
Okay. So if the question asker can re-ask it, would be good.
Okay.
Maybe they will. So yes, we ask to the question asker if he can specify what he means with the ALD, probably. But we will get back to this once we have a, better understanding. And in any case, we will have a few, maybe 10 minutes for general questions in the end, so we can get back to this. Thank you so much again, Professor Loscalzo. Thank you. This was a very nice discussion and presentation. Thank you. And now we will have a few moments of transition. So meanwhile, if the people in the audience want to have some coffee, and we will in a moment be ready with the next speaker. Okay, so after this short break, we are ready for the second speaker. And I'm very glad to have here and to introduce Professor Mariangeles Serrano from the University of Barcelona. Mariangeles is an ICREA research professor at the Department of Condensed Matter Physics of the University of Barcelona, and she obtained a PhD in physics at UB and a master in mathematics for finance from the Centre de Recerca Matematica, very good, CRM. She's a founding member of Complexidad, the Catalan network for the study of complex systems, and a promoter member of UBICS, the UB Institute of Complex Systems. Professor Serrano is a world-leading expert in complex network science, and her work focuses on the role of space in real networks, producing maps in a hidden geometry where distance measures the likelihood of interactions and enable multi-scale unfolding. She is also renowned for her research in multi-layer networks in which different types of interactions or nodes coexist. Applications of her network cover a wide variety of real systems, from biological to economic and socio-technological systems. We believe that this will be a particularly interesting connection. Mariangeles is really one of the leading figures of network geometry, one of the initiators of this whole field. So I'm very excited to have her here and to hear her presentation.
Thank you very much, Sabrina, and thank you very much, Guillermo, and all people from Algorithmiq for the invitation. I'm really very glad to be here and to share this time with all of you. So, as Professor Loscalzo was explaining, networks are not only in biology, not only at the cell level, but networks are ubiquitous, are everywhere. We find networks in society, in infrastructures, technology, biology at scales, in the brain, in the cell, and all networks talk a common language. And in our lab at the University of Barcelona, we are trying to understand this language. We are doing theory of complex networks to try to see how we can create methods and tools and techniques that allow us to understand the structure and the dynamics and the functionality and evolution of these complex systems that can be represented as networks. So basically you have complex systems made of discrete units which interact. Okay. So this is a very simple representation but very powerful. Now these mathematical representations of complex systems are helping us to understand real complex systems in a new way. They are like changing the paradigm of how we study complex systems, and at the same time are helping us to solve complex problems, and in particular are helping us to understand the multiplicity of scales which are present in complex networks and in complex systems. So typically when we have a network, we study a network, we take a single snapshot, we study the connections there, how the different parts of the system relate to each other, but we don't care so much about which are the scales which are present in our system. Are there different length scales, different time scales, different energy scales which contribute at the same time? And this is a central problem in complex systems. Typically in complex phenomena, there are a multiplicity of scales which are present at the same time, which are relevant, and which interact. So for complex networks, we have these questions. How are the multiplicity of scales which are present in complex networks, how these different scales interact with each other? This is related to other very central problems in networks, and I would say also in complexity science, like the problem of emergence. You know that one of the key properties of complex systems is emergence. Yeah? We don't have a theoretical definition, a canonical definition of what a complex system is, but we know a few features, and one of those, the most important one, is that complex systems present emerging behaviors. You know? And then this is directly related to the problem of scales, because emergence means that you have some phenomena at one scale that is related to phenomena at a different scale, and how this relation goes on is for the moment kind of mystery, and in fact there is a lot of discussion about the kind of emergence that we have in complex systems at the level of metaphysics. Is this emergence weak? Is this emergence strong? Do we need new laws at each level of emergence or not? So it's an important question that goes even beyond physics. It goes even beyond to philosophy, metaphysics, etc. This problem of scales, of the multiplicity of scales that collaborate to make emergent phenomena, is also related with another big question in science, which is the question of symmetries. So you know that in physical systems, and in particular in complex systems, there are sometimes invariance under transformations. You apply some kind of transformation to your system, and then you find that some pattern remains the same, some behavior remains the same. Yeah. So the thing is that this question in networks is very much related, as you will see, with the problem of scales. So what I would like to answer now, the question that I would like to answer now, is that which are the scales which are relevant in networks at the level of structure, and whether these multiplicity of scales which are present in networks are related to some kind of symmetry. To answer this question, we are settled in a framework which is network geometry. What we use to answer these questions are maps. We are constructing maps or complex networks. So you all know what a map is, and you all know the importance of maps in our everyday life. We use them continuously in our mobile phones to go from one place to another, to take just strategic decisions, etc. So what we are doing is to bring back a space to networks, because if we go back to this very simple representation that we are used to, this is a kind of topological object. In this kind of network representation, doesn't matter where the node is positioned, as far as it keeps the same connections to the same neighbors. So you replace a node in this topological representation in another place in this picture, and the meaning is the same of the structure. Yeah. Well, in a map, the place where things are positioned has a meaning. Yeah. So at the very beginning, when we were starting with networks, we were so happy that we didn't have geography, geometry, any kind of distance in that systems, because topological representations were very powerful in the sense that allow us to do a lot of calculations and to derive a lot of properties. While geometric things, geometric objects, are very difficult to treat, distances are introducing another level of complexity in our system. So at the very beginning, we were so lucky, no? But at some point, we realized that these structures, these complex network structures, are a small world. That means that every node is very close to any other node in the system if we talk, if we take as distances the shortest path lengths. Yeah, so there's a problem of separability. We cannot separate different regions of the network when we work with these shortest path distances, because networks are a small world. So we need to introduce geometry back to have this separability thing in terms of network structure. Yeah, and this is what we've been doing for the last 15 years or so. We've been trying to map networks, to try to relate the topological structures that we observe with an underlying geometry in which we can separate the different regions that are connected in the network in a geometric sense. Yeah? Now, our maps are a little bit different, are not the typical maps that we are used to in 2D, 3D Euclidean space. We use maps in hyperbolic space. This is an example. This is a map of the International Trade Network in 2013. And this is a hyperbolic map in which larger degree nodes are more positioned towards the center, while lower degree nodes are more positioned towards the periphery of this disk. And there are some other different issues that I'm going to explain you in the next slide, so you are going to understand what is the meaning of this kind of map. But just let me mention here that these methodologies, now we have models, we have also a mapping tool that allows to take as a network and to embed it into this space, into this hyperbolic space, allowed us to explore different kind of functions and attributes in complex networks, like, for instance, how navigable are these networks, whether it is easy to go from one place to another following paths into these geometries, to try to think a new way of doing community detection based on these geographical layouts, to understand hierarchies in a new way, to study robustness. So there are a bunch of things that you can do when you have the maps. And you can also use the maps to improve methods that are typically applied to the bare topology of real systems. Now, in particular, I'm going to focus here on what we did about the multi-scale structure of networks using our maps, using these hyperbolic maps of real networks. In particular, we developed 2 models, which are kind of paradigmatic because introduce a kind of renormalization group for the structure of networks. So I'm not going to talk about dynamics or anything like this. For the moment, we are going to focus on structure, because if you think about standard space, the space that we experience in our everyday life, to talk about the multi-scale structure is trivial. So you can see this room or a piece of this room, and the space is like self-similar to itself in a trivial way. In the case of complex networks, this is not true. So you cannot zoom in or zoom out into the structure of networks and see what is going on there in a trivial way. So we need a full methodology. We need methods and models to try to understand what happens there. And we came out with these 2 models, the geometric renormalization, that help us to understand real systems. So for instance, the multi-scale structure of the human brain, and the geometric branching growth that is kind of inverse of the renormalization group, that is allows us in this case to understand the structure of networks as they evolve in time. You will understand all this in a moment, because I'm going to a little bit deeper into these things, into these ideas. So let's start from the beginning. Let's try to see how do we define distances in complex networks. As I mentioned before, at the beginning, we were using shortest path lengths. Okay, the distance between 2 nodes is the minimum number of links that they need to go from one node to another node. But as I said, this has a separability problem. I mean, the collection of shortest path distances in complex networks is a well-defined metric, is a metric distance, but does not allow us to separate different parts, because the distribution is very homogeneous, let's say. If you take the whole collection of shortest path lengths in a network, this is a very big distribution around a very small value. So there is no way to separate things. Everybody is more or less at the same distance, you know? So it's difficult to separate things based on the shortest path length. So we came out with a completely different idea. Let's do the following. Let's couple the observed topology of a network with an underlying hidden metric space. It's like this picture that you have there, the iceberg. So imagine that you have the observed topology of your network at the tip of the iceberg, and then you have this underlying metric space under the surface of the sea that supports the structure that you observe. And the idea is that you have the nodes of the network positioned in specific locations in this metric space, and nodes are connected depending on their distances in the metric space, so that if you are closer in the space, you have more chance to be connected. And the clue for the connection between the 2 things, between the observed topology and the underlying geometry, is clustering. Clustering triangles in the network, because this is a kind of reflection of the triangle inequality that happens in the underlying metric space. Yeah? So this is the rationale, this is the idea, But then, how do you implement this idea? I'm trying to skip all technical details, but I have to say something about the most very basic model, because otherwise it's difficult to understand this renormalization group. To implement this idea of connecting the topology of a network with an underlying metric space, we came out with the geometric self-configuration model that we call SD in D dimension, but at the end is, I'm going to explain in 1 dimension. And it works in the following way. It's very easy to understand. You have nodes distributed in this underlying space, which in this case is Euclidean, is a circle. It's a sphere in 1 dimension. It's a circle. You distribute nodes at random there, so that nodes have a certain angular position, and you can define angular distances between pairs of nodes in this space. Yeah, we call this space a similarity space. If you are more similar to a neighbor, you are closer in this space. Yeah. Now you have a second variable attached to each node, which is the hidden degree, the kappa. The hidden degree, you can interpret this as a kind of importance of the node, a mass of the node, and the capacity to influence neighbors of this node. And then what you do, you have a heterogeneity of values. You can attach, I don't know, hidden degrees according to some power law degree distribution or whatever you like. And then what you do is to connect pair of nodes with a certain probability of connection, Pij there, which has this form. Yeah? This is basically a gravity law. It's nothing more than a gravity law. Or if you want, you can also see it as a Fermi-Dirac function. Yeah? But for us, let's say that this is a kind of gravity law in which the larger the masses, the larger the kappas, or the importances, the larger the probability that the nodes connect, and the closer they are in the similarity space, the larger the probability that they connect. Yeah? So we have this kind of gravity law model, the geometric self-configuration model, that we can use to produce topologies of networks based on a hidden metric space. Yeah? So these topologies are at the same time a small wall. That is a basic property of networks. You don't have this property. You have a network which is not a small wall. It's not a complex network. Okay? You can produce networks which are scale-free, yeah, which have high levels of clustering, yeah, etc., etc. Yeah? So it's a model that you can use to reproduce the typical topological properties of networks in the real world. You can use it as a kind of topology generator. Huh? Now, a very interesting property of the model is that it's isomorphic to what we call the H2 in 2 dimensions model in hyperbolic space. So we can go from this S1 formulation, this kind of gravity model in which we have nodes, importances, hidden degrees, plus distance in a similarity space, and a gravity law, like Newtonian kind of model, to another model, which is isomorphic. That means that we apply a little transformation to the hidden degree, to the kappa. We transform it into a radius. And we came out with a formulation of the model which is purely geometrical, but not in Euclidean space, but in hyperbolic space. So if we take the hidden degree, so basically the idea, we take the hidden degree, and we perform this transformation into a radial coordinate. That means that higher degree nodes are going to be placed toward the center, while lower degree nodes are going to be placed toward the periphery of the disk. We came out with a model where the probability of connection, so this transforms this probability of connection into this probability of connection, only depends on distances in the hyperbolic disk. Yeah? So we go out from a model that is based on similarity distance plus popularity, from a model which is popularity similarity and Newtonian, to a model which is kind of, I like to say it, Einsteinian, because it's purely geometric, just based on the positions of the nodes in this hyperbolic underlying space. Yeah? And then what is super nice is that we can go from one formulation to another because the models are isomorphic. So if you want to do kind of analytic calculations when you need to have the contribution of the similarity space explicit for your research, we can use the S1 model. But if you are more interested in something that is more related to geometry, yeah, you want to visualize, you want to have a picture, you want to navigate the system, then you go to the second version, and we can jump from one to the other as we need. Then to work with this hyperbolic geometry, it's not that easy. It's not that easy. Yeah, our everyday experience is in Euclidean space. Yeah, and there are some particularities in the representations that I show. What happens typically is that larger degree nodes are placed towards the center, while lower degree nodes are placed towards the periphery. Remember that if the degree distribution in networks is power law, that means that most nodes are low degree. So most nodes are going to be placed towards the periphery of the center. Yeah? Yeah? So is there any mathematician here? No? then I'm safe. The thing is that in hyperbolic space, in this kind of representation, the amount of space towards the periphery of the disk is larger, let's say. Yeah? So there is more space to adjust there more elements. So that's why the low degree nodes are more distributed on the periphery instead of the center. So the hubs are just a few and are closer to the center, you know. So at the end, hyperbolic geometry offers us a kind of natural space that is able to embed this hierarchical nature, this hierarchical structure of complex networks. Yeah, it's a kind of natural representation for the hierarchical structure of networks. So you have, I don't know, if in the S1 model, instead of using the circle, we say, no, let's use the sphere in 2 dimensions and we forget about popularity, or we take popularity as a second dimension in the sphere, there would be no way to include this hierarchical structure of networks into that representation. That's why we need the hyperbolic space there. Yeah. So we work in this kind of geometric models, which at the same time combine heterogeneity of degrees, popularity, and similarity between nodes, and where hyperbolic geometry emerges as the natural geometry to represent complex networks. You can use, as I said before, the models to replicate the structure of networks. You can just adjust the couple of parameters that the model has. There is one for controlling clustering. There is one for controlling the degree distribution, let's say. You can adjust these two values, and you can distribute, you can consider a certain distribution of hidden degrees, and in this way you can replicate the structure of complex networks, yeah? Which are small walls, but highly clustered at the same time. But you can also go the other way. You can take the observed topology of a real network and you can create the map. You can try to find the coordinates of the nodes in the underlying hyperbolic plane. We have now tools like Mercator, we call our tool Mercator, that you can use to produce this kind of hyperbolic representations of your networks right away. I mean, you can go to GitHub, you can download the tool, you give the tool your network, it produces the map, and some validation results to see that your map is high quality. Yeah. At the end, if you want, what this tool is doing is a kind of ultra-low dimensional reduction of the network. If you think that your network is an object of in Rn, let's say, in the Euclidean space in n dimensions, as computer scientists like to say, what we do is to have a representation in an ultra-low dimensional, in fact, 2-dimensional hyperbolic space. It's a kind of ultra-low dimensional reduction of your system. But that keeps information about your system. You are not losing that much information. And that's why we can do so many things with this kind of maps here. Now, once you have the map of your network, you can study different kinds of problems. You can study the robustness, hierarchies, community detection, navigability, but we focus on renormalization. Because we always had this question about the multi-scale structure of networks in the back of our minds, no? We wanted to understand what happens if we have the network at this scale. Okay, I measure clustering, the distribution, all the properties, statistical properties. What happens if I zoom out? If I change my resolution of observation, what do I see? Is it the same? Is it different? What happens there? Yeah, and we have tools in physics to do this kind of things in physical systems, which have been very extremely successful to understand important problems like the renormalization group. And I'm not claiming that we are applying a renormalization group here, because to begin with, we are just working with the structure of networks. We are not doing anything for the moment, about dynamics or function. But we take some ideas. To construct our machine that is able to zoom out on the structure of networks to see what happens there. And in particular, we took some ideas from the block spin model of Leo Kadanov, which is a precursor of the ideas of the renormalization group. And the main idea is that we will average over short range connections in our system to change the scale of observation to see what happens. And this is what I did. So in real space, if you have a lattice like the one that I have here, you need to perform a kind of two-step process to average over these short range connections. One is a coarse-graining, another one is a rescaling. Let's go first to this case, which is easier, and then you will understand immediately what we do in networks. So imagine that we have this lattice. Every color may represent a spin, the state of a spin in the system. And we perform a coarse graining. That means that we take every 4 boxes in the lattice on the top, and we transform into a new box here. Yeah, we do this coarse graining step. And then, in this case, we decide which is the color of the new box, depending on the colors of the boxes in the previous layer. Yeah, but this is not important for us because we only care about the structure, okay? But we do this thing. Now, the problem now is that this box is much larger than the box here. We need to rescale the box in these steps, so that's the size of the final box is the same as the one at the top, yeah? And what we did is just to take these 2 ideas. Now it seems very simple. When we were doing this with Guillermo, that was very, very complicated to realize how to do that. Now it's very simple. But we did is just to take these 2 ideas and to apply to complex networks. Once we have our maps, because here the key is how to define the coarse graining step. Yeah? Which are the nodes that we need to merge to produce a new node, a new super node in a coarse grain layer. Yeah? And once we have these representations in the S1 model, remember that this hyperbolic plane representation and the representation in the S1 model are exactly the same. So here you have a network embedded in the S1 model. Yeah? The nodes are placed in this ring, and then every node has a different size depending on the hidden degree. What we do is to make boxes of consecutive nodes in the similarity space to form a new supernode in the new layer. So for instance, these 2 guys corresponds to a new node here. And then we connect nodes in the new layer, whether they were connected, whether the nodes inside the supernode were connected in the previous layer. Yeah? And you can iterate. You can iterate this process to obtain different network layers. Yeah? This is easy. Now, what do you think that happens when we go to real networks and apply this protocol? We take a real network, we embed it into our underlying space, we produce this kind of network representation, the geometric representation. We do this coarse graining step of making new supernodes and connecting them and produce this sequence of layers. What do you think that happens? That real networks are self-similar. What you observe here are different statistical properties of the networks for different layers. For each network, we produce a multi-scale shell of layers, yeah? Each one which are reduced size, and in each layer we compute the different topological properties. So degree distribution, clustering coefficient, average nearest neighbors degree, communities if you want. You can measure different statistical properties in each layer. And when we represent these statistical properties as rescale by the average degree in the corresponding layer, what we find is that all the curves collapse. So the statistical behavior is exactly the same in all the layers of the multi-scale shell that we produce for a real network. Yeah? So we find this self-similarity in our system. This is interesting. This is interesting and can be very well explained by our models. Yeah? The only property that changes in this flow of layers, the only property is the average degree. The average degree of networks grow. Here you have the average degree for different networks and different layers, and you observe that the average degree grows and grows and grows and grows as we advance in the flow of layers. Yeah? That means that eventually, if we were in the thermodynamic limit, if we were able to reach a kind of thermodynamic limit and going to infinity in real networks, networks will arrive to a fully connected graph. But this is not important because for real systems, we will not arrive for sure to a fully connected graph. Okay? But just keep in mind that networks become denser and denser and denser and denser. Yeah. Yeah. What does it mean that there is a similarity in the network? Which is the conserved quantity here, let's say? Yeah? Here the conserved quantity is the probability of connection. This similarity can be explained because the probability that two nodes are connected remains the same in every layer. So if we change the resolution of observation of a network, there is not a change in the way nodes are connected to each other. Yeah? Now, what about something more practical? What about something more practical? Because at the theoretical level, that's very nice. We have this machine that allows us to change the resolution. Is there anything that I can use from a more applied way? Yes, we can use this to produce scaled-down network replicas of my system. Remember that I say that the only property that changes from layer to layer is the average degree that grows. Yeah? Then we can prune out this excess of connectivity in my system to produce a network that belongs to the same ensemble, that is produced with, let's say, explained by the same model. Yeah? So this is a very easy way to produce a replica of your network with a scaled down size. Now, apart from this, it seems that this normalization group of the structure of complex networks is good at explaining the multi-scale structure of human connectomes. In a previous work, in collaboration with Antoine Allard, we had shown that hyperbolic geometry is more natural than 3D Euclidean geometry, which is in principle the geometry of the human connectome, of the brain, the natural geometry, the physiological geometry of the brain, where it has evolved, let's say. Hyperbolic geometry is more convenient to explain how the different regions in the brain are connected to each other. Yeah? It's more, let's say, more congruent with the structure of connectivity in the human connectome. Yeah? But not only this, that these models in hyperbolic geometries are good at reproducing the connectivity in the structure of the human brain and the human connectome, but also that the renormalization group, this multi-scale unfolding that we can produce of real networks, is also very good at reproducing the multi-scale structure of the human brain. So we were able to collaborate with Professor Patrick Hackman in Switzerland, who provided us with a couple of data sets of multi-scale reconstructions of human connectors. I think it was like 90 people at the end, and in every multi-scale reconstruction we had like 5 layers, so layers for the human connectome of 5 different resolutions. What we did is to compare the properties of these multi-scale connectons, which were reconstructed based on anatomical considerations, nothing to do with our models, with multi-scale reconstructions that we produce taking the higher resolution layer. Yeah? So we took this higher resolution layer with 1,000 nodes, we produced the multi-scale reconstructions here. These are a production, and then compare with the empirical data, which was reconstructed just based on anatomical information. And the results are amazing. Yeah? So we're able to explain and to reproduce the properties of these connectomes very, very well. The rich club and correlations, the distribution, many properties. Yeah. So this is nice because it's not only that we recover this multi-scale symmetry of real networks, it's also that we can explain the multiscalar structure, at least of human brain connectons. Yeah? Now, we can also go the other way around. What happens if instead of zooming out my system, I try to zoom in? What does it mean to zoom in? To follow the evolution of a growing network. Yeah? Can I reverse this geometric renormalization group to explain the growth of real systems which are self-similar? Yeah, that's another question. So there are at least a couple of real systems that are self-similar. We found evidence for the relationship between scientific fields. We use as a proxy the Journal Citation Network and the International Trade Network. We studied the evolution of those systems over very long time spans of 50 years, of 100 years, and we saw that their topological properties were maintained the same in all this period that we were studying. Here you have curves for different years. You see that the properties are basically the same. So we came out with this idea of, can we reverse the renormalization group that we know that produces a multi-scale shell of self-similar networks to replicate this self-similar growth, which is some kind of inverse behavior in real systems. And we came out with what we call the geometric branching growth process. Yes, there is a way to do that. It's based on geometry, it's again based on our maps, and it's also based on a kind of branching growth of the system where one node splits into many nodes in the new layer, and then you connect the nodes in the new layer according to our models. Okay. Now, since we can use this renormalization group to produce scale-down replicas, and the geometric branching growth model is kind of inverse, now we can use it to produce scale-up replicas of real networks, right? And now we have this thing. If you don't care about the more theoretical aspects of our research, about the self-similarity, about this problem of emergence, whatever, at least you can use these two models, geometric renormalization and geometric branching growth, to produce a sequence of scale-up and scale-down network replicas, and you can use them to study different kind of things, different kind of problems. All problems where network size is relevant. And there are many problems where network size is relevant. Yeah? Not only this, but also you can use them to study dynamical process at the multi-scale. Yeah? Because in many dynamical processes, not only one scale is relevant, but there are many scales which participate. Yeah? And then now you have the methodology, we have the techniques that allow you to play with the size of real networks. That's what it's important. Before, we were able to play with the size of synthetic networks that were produced by network models. That was easy, yeah, right? But real networks had a definite size. We could not do anything about that. But now you can play with the size of real networks. That's a lot of fun. Yeah, you can change it up and down, and not only to have one replica, but to have all the sequence from below to above, and you can try to imagine many things that you can do with that. Okay. Finally, let me acknowledge my collaborators in this work. Guillermo is here, but well, there are some more people, but I mean, it's been hard work of many years, but the results I think are worthy. Thank you very much for your attention.
Thank you very much, Mariangeles. Of course, I know some of the results you talked about, and I find them very interesting and fascinating, but I also find immediately several potential connections with the previous talk. So let's begin with questions, and we will start from the audience here, and then we will go online. Yes, Marco. Yeah,
I have a very naive question. since you were talking about, let's say, inserting a metric space into these networks. And then as a physicist, when you talk about metric spaces, I think of dynamics. At the same time, you were saying you were not studying the dynamics of a network. So is this metric space just a way of visualizing the networks on the hyperbolic space or something like that?
Well, it's not only a visualization, it's a way to introduce separability in the structure of networks. Because if you just stay at the level of the topology, due to the small world property, there is no way you can separate different parts of your system. So if you are able to make a map, then you are able to separate. When you are able to separate, you are able to do many more things, like this multi-scale thing. You cannot do this multi-scale thing if you don't have this underlying geometry. It's not only to visualize. You can go, for sure you can visualize, and this is important. To visualize system is important. Yeah. But you can also go beyond, because without the map, without this kind of map where you have different parts, where you have an ordering of nodes in the ring, which tells you which are the nodes that you have to merge in the coarse graining to produce the multi-scale structure, yeah, you cannot do.
So maybe I'm missing the key ingredient here, because for this replica method to get renormalization, I can think of the Ising model. And in statistical physics, you do, I think, more or less something very similar. I guess you were also inspired by this. But there, I don't think you need a metric between the sides or
you know what happens? Yeah. The thing is, in the standard renormalization in physics, you don't need to care about the space, because the space renormalizes trivially. So when you are making the boxes, yeah, in this lattice, the larger box is trivially similar to the previous box, to the box in the previous layer. In networks, this is not the case. Yeah, you need this special ordering to merge nodes in this special way, yeah, so that you recover this similarity of the metric space. Now that we know how to make this zooming out of the system in a similar way, like we do in physics with lattices, now we can start thinking about dynamics. What happens if on top we put dynamics? There is a dynamical process on top or not. There is a dynamical process running on the network, and we try to see what happens if we zoom out. But what happens to the dynamical process? This is a step 2. But before, we needed the renormalization of the structure of the network.
Okay, thanks a lot.
So do you imagine that you could use it, for example, to study navigability in the network? Could it be an interesting or possible?
Yeah, but in the long, I mean, that this is going to be a very difficult problem. I think if, for instance, I don't know, epidemic spreading or any other kind of dynamical process there, yeah? If I look at the process, I see that some phase transition going on at some value of the parameter in finite systems or wherever. What happens if I go here? No?
Yes, yes. Matteo is arriving.
Thank you. It was a wonderful presentation. I understood part of it, so that must mean you presented very clearly, so thank you.
Sorry.
So I wanted to ask, as I heard your presentation, I began to think about some of the networks of networks in biological systems. And the problem we struggle with is, let's say you have a network in which you have your loosely connected protein binding partners in the periphery of the hyperbolic disk, on top of which there's another network that regulates the expression or the consequences of those. So for us, trying to sort out the issue of importance, however we want to measure that, becomes really paramount. And I wonder what you've been thinking about in terms of hyperbolic disks representing networks of networks that have interactions that regulate importance, and how that might be modeled, if you've done anything like that.
Well, if you talk about how this can be translated into the context of multi-layer networks, yes. I start thinking about this, but we will do it, but I don't have a clear idea yet of how to do it, because different layers can renormalize in some different ways. And then we need to find the extra connection, let's say, between because this multi-layer shell that we produce is for a single layer, let's say, in a multi-layer network. And then for every layer, you have its own multi-layer shell. So how are the two shells connected? Or this is not completely clear how to.
Yeah.
But for sure, yes, we will go also into that direction.
Thinking, what might be the features that define the movement of a node in the periphery more toward sort of centripetal features? What are how can you guide that process, and how do you quantitate the relative differences in hyperbolic plane localization as a consequence of the superposition of some regulatory element?
Yeah, we can talk later, because this is a very interesting idea. Thank you.
Yes. Matteo, wait, you go. Yeah, I have the impulse to go and give my microphone. I can. The volume is adjusted to my voice, obviously. She's typically very loud, so it only works for me.
I was just curious about this brain network that you were looking into. So the data was only for healthy brain, I assume. It was kind of like healthy individuals. Do you have any idea if there was some kind of dysfunction in the brain, how this would influence how the brain would look like?
No idea. It's something that we will look at very soon. We are collecting the data now.
Okay.
Yeah, I feel also curious. I have no idea. Maybe the similarity is lost.
Exactly. This was kind of whether there is.
Yeah. There are so many questions. This is starting. This is so. What about the meaning of cell similarity in biology? Why? Because of simplicity? Because communicability is better? But this is starting, kind of a starting point for something. But for sure, definitely something we will look at. Thank you.
Thank you.
Are there questions from the online listeners? So Boris, can you read them?
So yeah, there's a general comment from Daniel Cavalcanti. He's saying that he just wants to make a connection between your talk and the quantum community. He's saying that geometric networks appear natural in quantum communication because the photon transmission on optical fibers decays exponentially with the node's distance. And there are very few studies about this as far as he knows. He thinks that it would be interesting to investigate this further because the first quantum networks are being built right now.
Absolutely. And I know his papers. I know his couple of very recent publications about this quantum internet. And it's something in our list. So yes, we were thinking about trying to see. I'm not sure we can take these models right away into his quantum internet context, but maybe something very similar. And in case we come out with something interesting, I will contact him for sure. Yes.
Right, thank you.
Yes, of course, I can add a comment. Perhaps we do not know yet whether which are the laws of emergence of quantum networks, and whether they are different with respect to classical networks. Sure. So maybe whether you can apply, or whether they show the same self-similarity or not, is something that one doesn't know yet, but would be certainly interesting to study.
Mm-hmm.
And if there are no further questions.
There's also actually one more question. Yes. From Marilu. She's asking about, she's saying that it's a great and inspiring talk, and saying thanks, and is asking about dynamical versions of versions with dynamical networks, I guess, of this method.
So what we are doing now is to investigate dynamical processes that take into account more than one scale at the same time. So very simple process like the Butter model. So we take the Butter model, we run it on a network at a specific scale, and we're incorporating information from other scales. Like I'm a guide, I'm imitating a neighbor, and I take into account the position of my family, of my friends, of wherever, and I add this information into my decision, for instance. This is kind of things that you can do right away. This is kind of easy. We are working on this, which is the easy part. The renormalization of dynamical processes, this is the difficult part. Mm-hmm. This is going to be a kind of long-term thing, but we are going to start soon.
Thank you.
So thank you very much. I think we will have now a short break, and then we will go towards the some ideas about quantum networks this time. So we will explore a direction that is a bit different, because quantum networks are different. But before, we would like to thank again for the great talk and very interesting discussion, Mariangeles. Thank you very much. So a few minutes, maybe 2 or 3 minutes break just to change the presentation, and if you want to have some coffee. is more shifting towards quantum. And I am very happy to introduce here Professor Antonio Acin, Tony. And Professor Acin is also an ICREA research professor, but this time at ICFO. I don't know how to pronounce it. Is it ICREA? ICREA professors are really very prestigious professorship in Spain. It's actually one of the most important recognitions. So I'm really happy to have two of them here in our symposium. Tony has a degree in physics from the University of Barcelona and in telecommunication engineering from the Universitat Politécnica de Catalunya. He got his PhD in theoretical physics in 2001 from UB, and after a postdoctoral stay in Geneva, he joined ICFO in 2003. At ICFO, Antonio Acin leads the Quantum Information Theory group, and he is a world-renowned expert in quantum information theory. This is, in general, a very highly interdisciplinary area, combining tools and concepts from mathematics, computer science, physics, and engineering. He is one of the inventors of device-independent quantum key distribution concept. This was a milestone in the field of quantum cryptography, and he has also contributed significantly to research in the novel fields of complex quantum network. His research activity also covers aspects of foundations of quantum physics, quantum thermodynamics, many-body physics, and quantum optics. Thank you very much, Tony, for being here, and I am looking forward to your presentation.
Okay, thank you very. Is it working? Yes. Okay, thank you very much for the introduction. It was very nice. Yeah, so I'm here to tell you about quantum networks. So yeah, so I'm going to give you my standard talk at the moment about quantum networks. So I didn't shape my talk to the symposium also because I didn't know what the other speakers were going to present. So there are going to be some connections, maybe not as strong as you could think of. I'm not going to talk about quantum computers because we are starting activities in my group on quantum computation at the moment, but this is not what we do. I mean, and it was mentioned before, like in principle with a quantum computer, you could potentially, I don't know how, but you could potentially solve some of the computational problems that appear in previous talks. So in my case, the reason why we are considering quantum networks is because, well, so far many things in quantum, I will be talking mostly about quantum communication, but there are many protocols in quantum information in which there are two users, no, Alice and Bob. Okay, so if you have heard talks about quantum information, you have seen always Alice and Bob. And usually what happens is that you have Alice who prepares states and sends these states to Bob, and Bob measures these states. Or there is a source that prepares a state and sends particles to Alice and Bob, and they make measurements on this state, and they get results. Okay, and then you try from the statistics that you generate in this process and making knowing the states you prepare and the measurements you implemented, and same here, the state you prepare and the measurements the source prepared and the users implemented. Then you try to you generate some statistics of the outputs given the measurements that we call the inputs, and these are the measurement results. Then from the statistics you generate, okay, so you see here the probability of getting result B when you prepare the state X and you make a measurement Y, and here is the probability of seeing result A and B when you prepare the state rho or the source prepare the state rho and you make measurements X and Y. You try to make a protocol in this configuration, okay? And so far there were many results in quantum information theory based on this configuration. But what happens now is that, technologies are advancing, and now it makes sense, at least especially for me, I'm a theorist, so I can imagine about, say, near-term or close to near-term devices. And it makes sense now to start thinking about what you can do when you have slightly more complex networks. So I'm not going to be in the many-body super complex regime with many nodes. So I'm going to be, say, more modest, and I'm going to consider small networks in which, okay, potentially we will also want to go in the asymptotic regime, but at the moment I'm interested in small networks in which you have observers who share quantum states, and in these quantum states they can do some operations, so they have the possibility of making some quantum logic. They can make some operation on these states, okay? And then they have say you can produce single photon sources. They can share many states, so this means that there are different sources sending states, and then you can store quantum information in quantum memories, okay? And you can do some small quantum processes. So, okay, I think at the moment, I'm very interested in understanding what you can do with these network geometries. Okay, so you have these small networks, and we want to understand and go beyond this standard Alice and Bob two-party configuration scenario and understand what you can do. Okay, so this makes sense, I mean, theoretically, and also makes sense taking into account the progress in quantum information technologies. And also something that I like is that in this, because this is also one of our motivations in my group, so it's nice because you can connect, say, applications of quantum information with results in quantum foundations, and you will see some of these things in my talk. Okay? So you can think about the foundations of quantum physics, so say very speculative questions in quantum physics, and at the same time, with the same language, you can try to design new quantum information protocols that may find an application. Okay, so this is, I would say there are many people in my community working along these lines, so there are many people thinking about what you can do in these networks. In my case, I'm also interested in doing it in what is called the device-independent scenario. Okay, so this is a bit, maybe you haven't seen it, or maybe I mean, if you saw some of my talks, probably you have seen it, but I guess many of you didn't see any of my talks in the past. So basically, I want to model networks in some sort of very minimalistic way. So there will be a process sending these quantum particles to the observers, and the observers will take these quantum particles and they will make some measurements on these particles and they will get some results, okay? I will model this process of taking the particles coming from the source, making some given measurements and getting some measurement results as a black box, okay? This black box gets particle, you decide to make some measurements and you just put a label to these measurements. You don't attribute, you know it's a quantum measurement, but you don't know what's going on in the black box. And as a result of your action on the incoming particle, you get a measurement result. And again, this measurement result is just a label. Okay, so you can think of this as if your device was a box that you got from a provider. And this box, you have n buttons. And then you can decide to press button 1, button 2, button 3. And this is what you label with this X. Okay, so you have a box like this. Okay, this is not the practical way of doing it. But if you want to see it pictorially, what I mean is that we always reason, observers, we always reason with macroscopic actions, okay? I do an action on a box, or I put a knob in a given position. Okay, this is what I label by here, okay? So I press button 1, button 2, button 3. And inside the box, there is a process, a quantum process, that takes my choice and maps this choice with a quantum particle and produce a measurement output, okay? Also, observers, what we see in a quantum process, we don't see the wave function or any quantum stuff. We see the result. Okay, and the result, you can think of my box with these buttons that I can press. And then you can think at the bottom of the box, there are some lights. And when I press one of the buttons, one of the lights is on. Okay, so I interact with this classical way with my quantum box. Because again, we observers, we give classical information and we get classical information out of a quantum process. And at the very end, so there is a network that produces these particles. These particles go to the observers, and the observers decide to make these actions on these particles, and they get results. And my description of my quantum network is very minimalistic. It's just the probability. Okay, what is the probability that I see a given result A1, A2, AI, AN when I perform action X1, X2, Xi, Xn? Okay, and this is my description of the network. I know that behind the network there is a quantum process that gives rise to these probabilities, but somehow I don't care so much as a user about what's going on. Okay, so then what I want to understand is, like, from a foundation point of view, is that what are the probabilities, the correlations that I can observe in a given quantum network, and how this is affected by the topology of the network. And then once I understand the correlations I can get, how I can exploit these correlations in the networks to do quantum information protocols. Okay, this is why I say that I connect quantum foundations to quantum information theory. So why do I like this minimalistic, say, black box language? Okay, this device-independent scenario, sometimes I even call it quantum information theory with black boxes. Okay, my boxes are not black, but you should see it. You don't see what is inside the box. Okay, you simply put an input and you get an output. Okay, I like it because of the following. So now, we start having first generation of commercial quantum information technologies. Okay, so there are companies who claim to have a quantum computer. Okay, and there are companies who have quantum cryptography or quantum random number generators. Okay, and also maybe there are not so many companies, but also in scientific labs, people make systems that simulate other quantum systems. And the reason why we get an advantage with these objects is because they do things that we cannot do with our classical computers. But they charge you a lot of money for this, okay? Because technologically this is very demanding, okay? So now imagine you are a user and you want to run a quantum computation, okay? And then the machine gives you a result. What do you do with the result? Is it good? I mean, you cannot simulate what the quantum machine does with your classical machine, okay? So somehow you have to trust. And then you can go to the provider and say, or you want to run cryptography. Right? And then again, you run a protocol and did it work well? And of course, the trust you have is like you go to the provider and say, is this a quantum computer or is this a quantum random number generator? Is this really quantum? And the provider will tell you, yes. Okay? Well, because this is what you get when you go to the provider and you pay a lot of money. You get a box. Okay? So this is why I like this language, okay? Because it gives you ways to reason in terms of boxes, because at the very end, this is what you get from a provider. Okay? So if you can, opens a way of certifying also boxes, okay? So this is a problem that we care a lot in my group, okay? Quantum certification, okay? Is this really a quantum computer? Is this really something that I cannot do with my classical computer? Is this really generating randomness with a quantum origin? Is this really quantum cryptographically secure, okay? And I want to do this from the outside, okay? I don't want to open the device. Even if I open the device, I won't understand what is inside the device, okay? So I have to arrive to at some conclusions about the usefulness of these devices from what I see from the outside, from the statistics they generate. Okay, this is why I care about this language. And okay, I mean, if you have seen my talks, you have seen this joke. Actually, I'm always tempted to remove this joke from my presentations because I've been using it a lot, but I still like it, okay? Because it really represents what's the problem here, okay? So this is you, the user, and you go to the provider and you want to buy a quantum random number generator, okay? And then this is the problem, okay? So this is, the provider is going to charge you a lot for this quantum random number generator because it's quantum. But then you may wonder, okay, how do I know that this is quantum, okay? Because you know that the probability that a proper quantum or classical random number generator gives you this sequence is the same as any other sequence. Okay, so how do you know that this is quantum? And this provider tells you, well, the problem is that with randomness you can never be sure, but actually in quantum physics, and this is something that we proved already more than 10 years ago, quantum physics you can be sure that things are random. Okay, so this is a certification possibility that appears in quantum physics, and it's impossible in classical physics. Okay, but more in general, this is what I want to do, okay? So how can we certify quantum devices from the statistics they produce? Again, you don't want to open this quantum device and see how the outputs are produced, because actually even if you open, you won't understand anything. Okay, so this is why, I don't know, when did I start? I don't remember now. Okay, I still fine. Okay. And how much time do I have? I mean, I will stick to the time. I just want to know. 30 minutes, more 30, half an hour. Okay, good. Yeah. Okay. So this was sort of introduction to explain the scenario and the context that I care. Okay. So I care about this. I want to start putting complexity in my topology and understand things that go beyond the point-to-point configuration of Alice and Bob. And among the many possibilities, I want to consider the situation in which I only care about the correlations that my network generates. And I want to do reach conclusions about the performance of my network only from the correlations that are generated. Now, a bit for the experts. If you want to do things interesting for quantum information purposes in a network, the correlations you see, you should not be able to explain it with a classical model. Because if you have a classical model explaining the correlations, you cannot reach something that is impossible in classical information theory, and we, quantum information theorists, we want to achieve things that are impossible in classical information theory. So this means that these correlations do not need to have a classical analog, and this is called Bell non-locality. Okay, so you have to find correlations that do not have, or probabilities, say statistics that does not have a classical analog. In all my talk, there is an implicit assumption, which is these processes are local, okay? So the process of generating the output given input is local and does not depend on what the other parties do. Okay, so this is crucial assumption. Okay, what happens in my lab depends on what I see in my lab, and not, if, say, I'm here in Barcelona and Sabrina is in Helsinki with her box, what I see in Barcelona does not depend on what she does in Helsinki. This is a crucial assumption. Okay, so what was it was mentioned in the introduction. how did we arrive to that? Okay, so we arrived to this device-independent approach, not only for randomness, but actually we arrived for that because of quantum cryptography. So if you read anything about quantum cryptography, sometimes people, especially in the past, and I also made this mistake, people were claiming that quantum cryptography gives you unbreakable security. Okay, so then companies were selling you this, and you had unbreakable security. But then, in already more than 10 years ago, there were several teams that went to some of the companies selling quantum cryptography. They bought the devices, and they hacked the devices. Okay, so then someone was lying, or something was going wrong. Okay, because it was unbreakable, but these people were hacking commercial devices. Okay, so this is Vadim Makarov, who was the last author, and who were doing the first experiments on this. Okay, so how did they manage to break the quantum devices, the quantum cryptography devices? Okay, so you have to understand, when people say that they are unbreakable, they say so because there is what is called a security proof telling you that the protocol is secure. Okay, so there is a mathematical security proof. People construct devices based on this mathematical security proof, but then other people hack these devices. Okay, so there is something that we are missing. Okay, I think the good thing about hacking devices is that you learn from hacking attacks, is that you learn from them. Okay, and I think any cryptographic field is not mature if you didn't have hackers. So Makarov was a good hacker. He hacked the devices, but he collaborated with the providers to improve the security of the devices. But how come they hacked the devices? Well, what we understood is that the hackers broke the implementation, but not the principle. Okay, so what do I mean by this? Okay, so if you go to a quantum cryptography protocol or many quantum information protocols, you have a theoretical description. It tells you, I mean, for those who know quantum physics, they tell you you have to prepare these states in this vector space, and you have to make these measurements. Okay, so this is the theory, the theoretical description of your protocol, and this is where you prove security. Okay, this is my field theory. But now you go to the provider, okay, and now the provider, you tell the provider, you have to prepare states in a Hilbert space of dimension 2. Well, I mean, but this is impossible, okay? I don't think anyone can guarantee that something lives in a Hilbert space of dimension 2. So what people do, they prepare states with photon, okay, or with an atom or whatever. So an atom, a photon, is not something of dimension 2. It has a frequency, it has a polarization, it has, I mean, we all have many degrees of freedom, okay? Nothing, we are not two-dimensional objects. And then, and photons are not two-dimensional objects, and atoms are not two-dimensional objects, okay? So this assumption is effectively impossible to meet in practice, okay? And also this measurement. So the provider, what the provider does, well, provider uses laser sources and photodetectors trying to meet these requirements, okay? But I mean, there is always a gap between what happens in the implementation and what the theory demands in standard cryptography. So you have a security proof here, but the hacker somehow doesn't care about the security proof because the hacker is interested in hacking the implementation, okay? And you have a security proof here, but you don't have a security proof there. So how do you solve this gap? Well, just remove all these assumptions in your implementation, okay? So if you don't demand that you prepare states in Hilbert space or in dimension and you make these measurements, if you have a protocol only based on the statistics you generate, then there is no mismatch between your security proof and the implementation, okay? So this is another motivation for us to consider this device-independent language. Okay, maybe it was a long process just to tell you that we care about networks of quantum black boxes. I could have said this from the very beginning. Okay, so this is what we want to do. Now, okay, so let's now consider the simplest network. It's something called bilocality that was introduced in this paper that goes beyond the point-to-point configuration, okay? So now instead of having Alice and Bob who receive particles from a source, I make the you see, my networks are very simple. I mean, compared to what was discussed by Mariangeles, she was putting many nodes and things like this. We are not at that point yet, okay? So, or at least not for the type of applications I have in mind. Some other people may have other applications in mind, okay? So what is the next step? I mean, let's add complexity. We put not one source, but two sources, okay? So one goes to Alice and Bob, another one going to Bob and Charlie. Okay, so what is the things you can expect here? Well, if you know about quantum physics, there is a state being prepared here, times another state being prepared here. They are independent. A measurement here, times another measurement here, times another measurement here, and you combine these things with something called the Born rule, and you compute probabilities. So this is my description of the network. So I would say that some correlations are quantum whenever I can find states and measurements that I combine as a quantum physics textbook tells me to produce the observed correlations. So what are the classical correlations that I can expect in this network? Okay, sorry, you can remove this. Well, I have some instructions were prepared by this source, some other instructions were prepared by this other source. Okay, I'm assuming that they are independent because of the network topology. And then this output is produced here depending on the input and the particle coming from this source, you see here. Here, this box produces the output given the input coming from both sources because of the network topology. And this particle produces the output given the preparation by the source and the input. Okay, so this is the mathematical definition of my objects. These are quantum correlations, and these are classical correlations for the same network topology. And I want to see when an object that I can get with quantum theory does not have an analog in classical theory. Now, maybe you know that. The nice thing is that this network topology connects with something that was invented before quantum physics, which is the language of causal networks. Okay, so maybe you are familiar with this. So in causal networks, you observe some correlations. You want to understand the causes behind these correlations. Okay, so a typical example, no, is like you see that people who smoke tend to have cancer. These variables are correlated. So what we say is that if you smoke, you have cancer, causal connection. Okay, but only from the correlations between these two variables, you cannot exclude that if you have cancer, you tend to smoke. Okay, it sounds like crazy, but from the correlations, you cannot exclude these two things, okay? And actually, tobacco companies, they may tell you, no, there is a variable that you didn't monitor in your study, which is, I don't know, that some people are more nervous than others, and people who are more nervous, they tend to smoke and they tend to have cancer, and this explains the correlations. Okay, so you see correlations among variables, and you try to explain this with causes, and the way you represent this is with arrows, showing the causal connections between your variables. And this is not invented. I mean, there is a field studying this, and this type of considerations are used in many disciplines. I mean, I believe in medicine for sure these things are used, okay? So what is the language these people use? They use the variables they use the variables you observe, like people who smoke and have cancer, say, with a circle, or well, you can choose in a given form, okay? Then they represent the causes with arrows, okay? And sometimes they believe that there are some other variables that are not monitored in the experiment, these hidden variables, but that may be behind and they may explain some of the correlations you have, okay? Like, okay, I didn't check in my study whether people are nervous, but I can think of people being nervous and creating this, okay? So these are called hidden variables, and you put a different symbol for that. This was invented before quantum, okay? So this, okay? So the nice thing is that all this language approach, all this language is useful for causal network, but it's also useful for quantum classical networks, okay? So the standard network that people study with Alice and Bob, getting particles from a source and making measurements, you can represent with this causal network, okay? So you see, this is the state prepared by the source. It's hidden. I don't have access to it. And this goes, and in my location, I choose to implement a measurement and getting a result. So my choice of measurement is a variable that I have access to, and I get the result. And this output depends on what I decide to measure and the incoming particle. Okay, so you give me any network in this black box approach, and I map it into a causal network. And you give me any causal network, and I map it into a quantum network. Okay, so there is a very nice connection between these two things, okay? And, well, it could be that these are classical instructions, or it could be that these are quantum instructions, okay? An entangled experiment is the one in which these are quantum instructions coming and producing the output based on quantum information, okay? And standard Bell theorem experiment in which you prove that some correlations are possible in quantum physics and not in classical physics. It's simply a situation in which the network topology is the same. You simply change the nature of your particles from classical to quantum. But the network topology is the same, the causal network is the same. So what does it mean then? Bell theorem is, again, the theorem that tells you that there are some correlations that you can get in a quantum network that cannot be explained by a classical network, by they don't have a classical analog. So if you reinterpret this theorem in terms of causality, this is telling you that there are some correlations that you can observe that you cannot explain with a given causal network if you have access to classical information, but you can explain with the same causal network if you replace classical information by quantum information. Okay, so say classical causality cannot explain some correlations for a given causal network, and quantum can. Okay, so now, in principle, something that we are interested in is like, I mean, there are books on causality, okay? There are books like this, where they tell you how to do causal network studies, okay? These books only care about classical causality. And what we know, because of Bell theorem, is that quantum causality is different. So, in principle, here there is something that we are interested in. Let's try to understand how causality is affected by quantum information. So, I think this is maybe speculative at this moment, but if one day we think that there are quantum effects in biological processes or medical processes, okay? I believe this, I'm not an expert on these things, but I believe these causal networks are used already in classical processes in biology and medicine or thermodynamics or whatever. So at that time, if quantum effects play a role in these processes, we'll have to understand how quantum causality behaves. And at the moment, we know that this is different, okay? Because here is a toy model in which there is a difference between classical and quantum causes. Okay, so this is one of our motivations. So, okay, I'm talking a lot. Usually I talk a lot in my talks, okay? But I started with a motivation from a quantum information perspective, this certification. This is why I care about black boxes. But then in this process, we realized that there is a connection with causality, and we understood that quantum causality is different from classical causality, and this is why we also care about this. Okay, so now, again, we want to characterize quantum correlations in quantum network, which means we want to understand how causality is affected by quantum information. Okay, it's the same. One more causality oriented, the other one more network quantum information oriented, but it's the same question. I mean, this is a hard problem, okay? So you can look at this book, and there are many results there. So this is, again, if I give you this simple network, okay, where are these causes producing these correlations, I mean, what is the set of correlations I can see? Well, it's a hard problem because the sets you get are non-convex. Okay, so I'm not going to give you a talk about this. Just believe me, this is hard. Okay, so we got some results on this, and we saw how these objects, which is the set of classical correlations, are vary when you have quantum correlations. Not easy. Okay, and actually, this is also a complex problem because the methods we have, even if we are very happy that we got some methods, scale very bad. Okay, so as soon as you have a large network with different causes, it's very hard to understand, characterize these objects. Okay, so I think this is a challenge. We shall see. Okay. Good. Yes, so now, I told you that on the one hand, we need to characterize these correlations, and this is difficult. We got some progress, but we are still far from having a good understanding of this question. Now, this language of the networks also allows you to understand very speculative questions in quantum physics. So maybe this is a bit speculative, well, a bit foundations for this audience, but okay, maybe it's a result that I'm proud of, so I will explain it anyway, okay? So basically what we did, we posed a question about quantum theory, because if you ever study quantum theory, usually there is a very standard formulation of quantum theory, which is in terms of Hilbert spaces. So if you have a textbook in quantum physics, you open page 1, there are the postulates of the theory, and the first postulate says, to any physical system, you associate a complex Hilbert space. Okay, this is the first postulate of the theory. Now, I don't know if you ever thought about this, but it's a bit weird that you have a complex object to explain physical theory. Okay, so for me it was a bit weird to find this. Okay, because, okay, so this is what I was telling you. Okay, so if you open a textbook in quantum physics, you have some postulates. Okay, well, for those who know them, well, you have them here again. For those who don't know these postulates, just a mathematical formulation of the postulates, page 1 of any textbook. But I think what is a bit weird is that you express everything in terms of complex numbers, not real numbers. And actually, it's the first theory where something like this happens, okay? And I mean, okay, I'm a poor physicist, but even the founders of the theory were a bit worried about this, okay? So Schrödinger wrote to Lorentz saying, well, I mean, how can it be that we express things in terms of complex numbers, okay? Our reality is based on real quantities and real numbers, okay? We don't see complex numbers. So what happens if we replace complex numbers by real numbers in quantum theory, okay? So you take the postulates and you only make this, you see? I mean, the only difference is like complex, real. Same structure, but I replace complex by real numbers, okay? Do I get the same theory? Because maybe it's more satisfactory, okay? So, I mean, this was considered by others, okay? And first, I want you to think that before quantum physics, complex numbers were never used in any physical theory. So if you think about electromagnetism, we use complex numbers sometimes, but this is only, we do this only to simplify calculations. I mean, the Maxwell equations, there are no complex numbers. Okay, we sometimes use to combine the electric and magnetic field into the same field, and we use complex numbers, but there is nothing. The complex numbers don't play any fundamental role in the theory. Okay, so quantum physics was the first theory in which complex numbers played a role, but we wonder, okay, maybe you can remove them and you have another theory, okay? Or maybe the same. Do we really need them? Okay, so we were not the first considering these things, okay? And I'm not going to explain this, but there was a previous paper in which people showed that if you have a network in which a state is prepared and different measurements are prepared on this single state, you can explain everything in standard quantum theory with real numbers. Okay, so there is no gap between complex and real quantum theory in a standard Bell test. So in a way, this paper seemed to close this discussion. So what we proved in this paper is that no, if you go to a network, already this network that I show with two independent sources, you can see a gap between complex quantum theory and real quantum theory. So now you can make an experiment and falsify real quantum theory, hence understand that replacing complex by real numbers in your standard formulation of quantum theory makes a difference. Okay, so you could argue that you need complex numbers. Actually, you never prove that you need something in a theory. You falsify things, okay? So strictly speaking, you can falsify real quantum theory. Okay, and for that, you need to exploit network topologies, because if you do it in the standard Alice-Bob configuration, you will never do that, as these researchers proved in the past. Okay, so this is a situation in which we really had to exploit the concept of a network, a very simple network, to achieve a result that was impossible before. Okay, so what about applications? Well, this maybe is a bit technical for this audience, but I will try it anyway. Okay, just to give you a situation. So one of my obsessions is how to make these device-independent quantum information protocols I was telling you before. Okay, so in principle, you want to have protocols with these black boxes. They are very nice because they are self-certified. You don't need to put any trust on the provider. Either you see statistics and then you are happy with these boxes, and you don't see the statistics, you give them back. You don't need to open them. So what is the problem with this? For instance, if you think about cryptography, okay, there is Alice, Bob, or there is Sabrina in Helsinki, I'm in Barcelona, we want to exchange a secret. And we want to do it in this device-independent way. We want to do it only from we want to conclude that we have secrecy only from the statistics we see. So for that, we need to generate statistics that does not have a classical analog. Again, because if I can explain the statistics with a classical analog, I cannot obtain a result that is impossible in classical information theory. Now the problem is that it's very difficult to generate statistics that does not have a classical analog at distance. And she's in Helsinki and I'm in Barcelona. Okay, so they should, Sabrina and I, or Alice and Bob, they should observe a Berlin inequality violation. They should prove that these correlations do not have a classical explanation, and they have to do it taking into account that they are distant. So what is this difficult? Okay, no, this we don't need to say. Well, it's difficult because we are connected by a channel, and the channel has losses. And if you want to do quantum information quantum communication through a channel, you have to send light at the single photon level, and a single photon gets lost in a channel most of the times. So we have to play, and we have to achieve the result, taking into account the losses in the channel. Okay, so maybe there's a bit, I will go faster here. So what is the problem then? Is that if I send a photon to Sabrina, the photon will never arrive, or will arrive with very small probability. This is connected to what Danny Cavalcanti was mentioning before. Okay, so if you try to model a network of quantum communication, you always have, or the standard network is made of fibers in which you send photons, and photons usually get lost. So it's a typical situation in which direct transmission, point-to-point configuration does not work. So you have to do it in a different way. So I don't have time. I don't think it makes sense for this audience to do that. So just for you to see that, then people came up with solutions. Okay, one solution is the one you have here. Okay, Alice and Bob send stuff to a central location, and depending on these outputs in the central location, they can run the bell test, and we can make security. Or, well, just for you to understand, I mean, I don't want you to understand all the details, but the setups get complicated simply because direct transmission is not possible. Okay, so this is another proposal by Gisèle Pironion-Sanguar. Okay, they have to send stuff here, they have to make measurements. We also made this proposal. You see, this does not look as a direct one-to-point configuration. Okay, it's more complex. If you're interested, I can give you the details of these things. I think it's beyond the scope of this talk. Okay, so this was our proposal. Okay, but this, I think it's time to conclude with this. Okay, so what did I want to tell you about? Okay, so I think now quantum networks are starting because we really have a first generation of quantum networks. Okay, there are experiments in which people produce 2-3 entangled states, and they send these 2-3 entangled particles to 4 or 5 locations, and they make measurements on these particles, and they get some correlations, they get some statistics that does not have a classical analog. Okay, so this is happening. So it makes sense, and it's timely to study what you can do with these networks. Okay, potentially in the long term, we will escalate this to larger networks, and we'll study perhaps asymptotics and things like this. I think it's even interesting to do it now. Okay, so there are the two ways. Okay, you study the more asymptotic regime that we will potentially eventually reach, or you do it now with the things you can do at the moment. Okay, so in my case, I work more on small quantum networks, but okay. So we already know that these small quantum networks, even if they are small, you can achieve a result that are impossible in standard point-to-point configuration. Okay, so I gave you an example, a fundamental example. So when you want to test real quantum theory, you cannot do it in a point-to-point configuration, but in a small quantum network, you can falsify real quantum theory, hence conclude that you need complex numbers in quantum physics. And I also, very quickly, because I think it was too technical for this audience, give you another explanation in which I was telling you that if we want to do this device-independent cryptography, in which you can see systems as black boxes, you cannot do it with direct transmission. Okay, you have to do something a bit more creative. Okay, so I think this is a very open question at the moment. Okay, so what you can do, understanding what you can do with quantum information exploiting network geometries, this is very open. So maybe the question will be not much, maybe a lot. We shall see. Okay, and this is why I'm here, I believe. Okay, so thank you very much for your attention.
Thank you very much, Tony. Really nice talk, and bringing us to another perspective, which is a perspective that goes more from quantum towards networks. So from some fundamentals to what could be in the future a potentially interesting technology. I have to immediately make a comment. I will take this opportunity about the fact that there are some points really that Tony mentioned that are extremely important also in the field of quantum chemistry simulations, quantum computing, and so on. And this important point is, number one, the fact that there is a distinction between what the theory says and what the implementation actually is on real devices. So we really need fundamental results like the one that you discussed, and fundamental approaches and perspective in order to make sure that what we are implementing in real noisy devices is actually what we know from the theory, and how can we adjust. So I liked it very much. And then, of course, personally, I believe that only by bringing together fundamental quantum physics, fundamental quantum information theory with the applied sciences, you can find a way to such complex problems. So I really appreciated these examples. Okay, ready for questions? Yes, Eric, wait a second.
Thanks for a wonderful talk. So a question on the or maybe a comment on the connection to causal reasoning of Perl. I remember that in Perl there is a lot of emphasis on on making experiments where you change the network. There's the do operation and the see operation in Perl. Do you see a similar role or an analogous role to quantum networks and quantum causality?
Yeah, okay, so I must confess that I haven't read the book by Perl, okay, so I use it in my talk. But yes, but I know that there are these do actions and things like this. So I'm not aware of any paper doing these things, but I think it's, let's say, a very open territory just to take what people did with classical information and trying to understand what will be the analog with quantum information. So yes, I think it's something that makes a lot of sense. Maybe since I'm not super familiar with the classical concept, I cannot tell you in which direction to go exactly, but I think since we know that things behave different, I think it makes a lot of sense to take the do action and change the network and see what happens then for quantum information in the same network, yes. At least I plan to do it in the near future, yes.
It's kind of a continuation just to what you said. Because I think the idea with the kind of do calculus in Perl is that you are able to, even when you cannot do a fully controlled experiment, so when you cannot do the do's and you only have these correlations, that you would have a way to, for example, exclude the possibility of having an extra variable influencing. And I think similar kind of ways of doing it quantum computationally could help you with the certification problem as well. Let's say that even if you don't have access to control certain parts, you could, with your correlations, exclude, let's say, the possibility of having something external influencing your outcomes.
Yeah.
Are there questions from the online? Yes.
So yeah, Aritra is asking about modeling this on the slide of the device-independent scenario, whether it's possible to model to design a framework which kind of models this P function as a reinforcement learning agent.
Okay, I'm not sure if yeah, okay, so this device-independent scenario, let's say, well, I mean, it can be anywhere, no? So basically, in this device-independent scenario, or maybe let me just put it. Let me just maybe here. So I think in the device-independent scenario, the way I see it at the moment is like this, okay? So you only care about the statistics, so you will put, say, any experiment, okay, what happens in any experiment, classical or quantum, is that you have some systems that are being prepared and you make some actions on these systems and you get some outputs, okay? So maybe it sounds like trivial to you, but this is, to me, the zero explanation of any, not even physical, any scientific or scenario, okay? So you just, you have some actions and then you get some outputs. And in the device-independent scenario, this is all what you do. So you don't put any learning process on that, or you don't put any computational assumption about what's going on. You don't say that this is a given physical system. You're not saying that this has dimension 2, 3, infinite. It can be whatever. Okay, so you only impose the variables you observe, the hidden variables, and the causal connections. That's it. And then you see the statistics that this network, potential network, generates. Okay, then, okay, there's a problem of causal inference. You may say, start from this and see if this can explain this correlation, okay? But if not, you can think like, I assume that in my scenario, this is the DAG, the causal pattern, and this is the correlation I see. And this, you don't have to assume. You make the experiment, and you see the statistics, you count frequencies, okay? And this is something that you see with your bare eyes, let's say, okay? Now, what you can do is like, you try to then see if a learning process could be here just to produce these statistics. But this is like putting finer details to these black boxes or balls, circles that appear here, okay? So this goes beyond the device-independent scenario. The device-independent scenario simply remains at this very minimalistic regime. There are no learning agents, there are no qubits, there are just particles propagating, inputs, outputs, that's all. Okay, so I think, of course, if you want, sometimes you want to do things like dynamics, you have to put some structure to these objects, and you then say, okay, I know that this is a learning agent, or this is a quantum computer with some given amount of qubits, and then this translates into more constraints on these correlations, okay? But in the device-independent scenario, you don't want to make any assumption about any of the objects, apart from the causal connections that you have.
Thank you.
Yes, one more.
Okay. One more question. What about using quantum causal tomography for inferring metabolic networks or gene regulatory network discovery?
Okay, I'm not sure if okay, first thing I want to say is that tomography. Okay, so let me just say something. So I work a lot on device independent, and my talk was on device independent, but of course, there are things beyond device independent, and standard quantum information theory is not device independent. So in particular, tomography is not device independent, because in tomography, you want to reconstruct a matrix, and a matrix has a dimension because it has a size. Okay, tomography is not a device independent concept, because if you say, I want to make tomography of this system, you are already providing a dimension for this system, okay? So when you give a dimension, this is not a black box. It's a system with a given dimension. So another interesting question, which I think it's completely unexplored. So if you think about causality, the way I quantize, so I was having this network, and in standard classical, everything is classical, and the way I quantize the causal network is I made the hidden variables quantum. Okay, so this is what I did, okay? So you see, I only changed the hidden variables and mapped them from classical to quantum. Another possible further quantization of causality is like you also make these variables, observed variables, quantum. Okay? But we don't, what we see, observed variables are never quantum, are always classical. So, but then to make these things quantum, okay, I don't know exactly how to do it, but maybe you have to put a dimension here and then do tomography. So it's no longer device independent, but it's a way of quantizing a network that goes beyond what I did, okay? So now you quantize these objects. These objects now become quantum systems with a given dimension, and you can try to see what are the, I don't know, quantum causality thing that you can observe here with tomography. Maybe this goes more in the direction of the question. But this is I don't know how to do it.
All right. Thank you.
So are there more questions? I do have one that is, so you were discussing before presenting the Makarov experiment and how somehow this mapping from a mathematical principle or a theoretical principle into the implementation was what generated the whole field of device-independent, device-independent many things actually, device-independent quantum information. But within this specific example, or more in general, there could be two factors that play a role. One is the fact that the moment you implement something on a physical divide, as you say, the Hilbert space is not the same, it's always an approximate implementation. And the other factor can be the noise itself. So they may be actually separated. One is really how you encode a mathematical object or an abstract object into a physical system, and the other one is then the noise that always affects these quantum systems. So are there in between, so they're not completely device, because a device independent take into account both in a way. So are there frameworks that are in between, so that they assume, for example, that the implementation is accurate and then they don't consider the noise, or the other way around? Is there a scope to do something that is not so extreme as device independent? So you put in some information in order to have perhaps easier access or easier, in a way, amount of information. The amount of information you would have is more if you add more details inside of what is inside the box.
Yeah, okay, so, well, first of all, your question made me think about something I didn't mention. It's like all this device independent or goes in the direction of quantum certification. So I think also for a technology to be mature, you need standards and certification, okay? And this is missing. So for instance, something that I find very frustrating is that I believe that there is something called quantum randomness that is different from classical randomness, okay? But now if you go to the standards for quantum random number generators, or the companies who sell you quantum random number generators, you say, okay, how do I know that this is random? And they tell you, because we pass all statistical tests, okay? There is a battery of statistical tests, and they tell you, we certify this is random because we pass these tests. But there are classical random numbers that pass these tests, okay? So we need intrinsically quantum ways of certifying quantum devices. Because if you certify something with a test that is passed by classical devices, then I'm not sure if I'm going to pay a lot of money for something that is certified with something. I mean, there is something much cheaper that passes the same test, okay? So I think, first, this quantum certification, we need intrinsically quantum certification test for quantum devices. Now, yes, of course, the device independent is like the most pure way of certifying because you don't make assumptions, and then somehow you could reason, especially for cryptography, the more assumptions you make, the weaker your security will be, okay? Because if one of your assumptions fails, you are in troubles, no? This is what happened in Makarov, no? So Makarov went to the company thought that they have a very good understanding of the device. Actually, in Makarov, what they did was reverse engineering. They bought the device, they opened, they understood the device better than the company, and this is why they hacked. So in the device independent scenario, this is not happening, okay? But what you can do, you can start putting assumptions, and then things become, because device independent is very nice, but it's very difficult, it's very experimentally challenging, okay? So you can start putting assumptions and alleviating some of the experimental difficulties. And I think in the long term, all the certification will be, you will have a piece of paper, well, I don't know, we shall see. But you will have a piece of paper and you will say, okay, these are assumptions I make about my preparation. Yeah. I think I'm preparing qubits, I think I'm making these measurements. And I'm charging you this. And the customer will decide whether it's happy or not with this.
Yeah, but definitely the whole field of certification for quantum computer, quantum devices, it's really important, and it's one of the major challenges nowadays. So I would say at this point we can thank again Tony for the fantastic talk and for the discussion. And now we will have the last speaker, Guillermo Garcia-Perez. Guille is the CSO, the Chief Scientific Officer of Algorithmiq. What he will do in his short talk will be to try to present what is for us a possible direction in the combination of the field and of the topics of quantum simulations, in particular quantum chemistry simulations, complex networks, and complex network medicine. So this is something that we would like to see as a new field which is starting and growing, and we have some ideas on what could be the potential enhancement provided by quantum for the field of network medicine. And along the way, we think that some of the tools that were created, of course, in complex network science, which is obviously was in the combination of complex network science and medicine and system biology, the whole field of complex network medicine emerged. But there are other aspects of complex network science that we would like to see more connected, like the network geometry, as possible tools to improve the predictions and to improve this discipline itself, which is very fascinating. And at the same time, as Professor Loscalzo mentioned in his last slide, presents several challenges that perhaps could be addressed potentially with the combination of these different disciplines. So let's see. You need a few minutes for a break? Yes.
I don't, but maybe people want to.
If do you want? No, maybe we go this time without a breaks, because this is a small talk. It's only 20 minutes, and then we will have discussions, and we continue after the discussion. I just want to. Because meanwhile we will fix the computer, like 5 minutes break just to fix the connection. Okay, so welcome to the last talk. This is a internal talk by Algorithmiq. So Guy, as I mentioned before, is our Chief Scientific Officer, and he will tell you a bit what we think is a very interesting vision and possibility for the future. That means combining complex networks, complex network medicine, and quantum physics in what we call quantum network medicine.
So hello everyone, and thank you very much for being here. So I would like to start by with a reflection about the current state of the art of different topics that we've heard about. In particular, well, what we've witnessed in the last years is that there is a drive towards this complex systems perspectives in science. So we've been driven beyond reductionism, which is a very big step, as we've heard. And network medicine in particular it's an embodiment of that in the applications applied to medicine. And given that human biology is definitely complex, that's very promising, and it has shown indeed many successful results. But at the same time, it's also a field that is computationally very heavy. It relies on managing, it requires managing lots of data and doing very heavy simulations. So in parallel, we're witnessing also the emergence of quantum computing. It's starting to happen only now. It was conceived many years ago, but it's becoming a reality only now. So it seems like a good idea, obviously, to join the two and see how they can complement each other. And we want to basically sparkle this discussion and show what we think could be very interesting points of contact between these two disciplines. So essentially, network medicine, please let me know if I if I didn't understand correctly how it works. But let's say one starts with a depiction or description of the system, a very complex system in terms of, for example, interactions between proteins and all the cellular components that might play a role in the biology of the disease. And by identifying some of these nodes that are known by other means, maybe reductionist means, are known to be related to the disease. Then in the neighborhood of that set of nodes, a module is identified, which might indicate other cellular components that might be related to the disease or play a role in the disease as well. And this is done at the level of a network analysis. By the way, I think could be a very interesting point of contact with the network geometry field, because in network geometry also the embeddings reveal this community structure in different ways, and also taking into account multi-scale structures. So it might be a very straightforward connection to explore. Then later on, that identified cluster of nodes is validated by other means to see if it really makes sense. So if those components really may play a role in the disease. And once this is done, we can proceed to understand how the disease actually works in a more mechanistic way. And also exploit that knowledge to do something useful with it. For example, identify possible targets that could lead to treating the disease, or at least to understand how to perturb that network in order to drive it into a different state. And then there is the question of, okay, even if that is done successfully, and there are several challenges, of course, along this path, but there is a question of, once the perturbation or the desirable perturbation on the network is identified, there's still the point that one needs to find a way to actually implement this if we want to have one wants to have a clinical impact, let's say. So one needs a drug that does this, a molecule, some sort of treatment. And that means that one needs to be able to assess or predict somehow whether a given molecule is going to interact with whatever it's supposed to be interacting with. So for example, with the proteins or other cellular components that it should bind to. And that no longer is really a problem of holism. It's a very reductionist problem. Given a protein, given a molecule, do they interact? That boils down to actually very, very fundamental theory of nature, which is using quantum mechanics, for example, to understand the behavior of the electrons in that system of molecule plus binding pocket, for example. So in the end, one needs to solve a problem that is rather reductionist at that level. And of course, it's not said that one cannot approximate this problem, for example, using classical methods, or also if one has information about those molecules beforehand, like because of experimental data regarding, because it's a known drug, for example, one can use this, and indeed that is how it's used for drug repurposing, because it's already understood what the physicochemical properties of that molecule are, physiochemical properties. But if once you do this from de novo, let's say, one needs to solve quantum chemistry problems. And that is usually a very important, a very big hurdle, because it's not easy at all. Actually, simulating quantum systems in general is quite difficult, because we are forced to working, unless one does it in a device-independent way, when one needs to actually model or simulate mathematical objects that have very, very large dimensions, like, for example, the quantum state and so on, that requires usually exponential amounts of memory or exponential amounts of time, and it becomes infeasible for even quite small systems. So actually, it's such a severe problem that this is what led physicist Richard Feynman in the early 80s, in 82, to conceive the idea of a quantum computer, that maybe we should rethink what we consider a computer. It's a physical system doing calculations, but maybe it doesn't need to be a classical system doing calculations. It can behave according to the laws of quantum physics, and as such, it can exhibit the same phenomena that makes the quantum systems that we care about difficult to simulate, and therefore solve the problems. So that was the original motivation for quantum computing. Later on, it was identified and understood that if one had such a machine, one could use it for other things beyond quantum simulation. One could factor numbers really fast, and other approaches. But quantum simulation, or simulation of quantum systems, lies at the very, very heart of quantum computation. And in this case, it seems like perhaps one of the most straightforward ways of combining this holistic approach with something that requires simulation at some point, let's say. But now the question arises, how far are we from achieving quantum computing? And of course, this is a bit of an ambiguous question, because it depends a bit on what do you mean by having a quantum computer. We already have quantum computers. So one maybe can ask, well, how far are we, in more like in a pragmatic sense, how far are we from having quantum advantage? Quantum advantage is, the name is quite self-explanatory, but basically means that we would be able to do, it's the ability to perform calculations that are impossible with classical computers using quantum ones. But one could think of different classes of problems. It can be problems that are actually not useful at all. It's just a proof of concept, just to prove the point that such problems exist. Or one could think of useful, in some sense, quantum advantage, meaning, for example, for the advances of science, understanding some specific quantum system that might not have any industrial impact or application. And then one could think also of practical quantum advantage, that is, for example, incorporating quantum computers in actual industrial processes. So currently, we are in this phase, which is already a lot. We've already achieved the proof of quantum advantage for not useful problems, but it is clear that this is a reality, that it's happening, and that very soon we will have useful and potentially soon as well practical quantum advantage. Now I can be a bit more precise or concrete about what, not so much in terms of the applications, but in terms of the devices themselves. So quantum computing was conceived soon after Feynman proposed it. It was formalized as a very specific machine, let's say, it's a theoretical model of a machine that would do a very specific set of operations. It would have a very specific set of properties, which we can call being fault tolerant. Fault tolerant means basically that we can rely on the machine, that errors are not expected to happen. This is what happens with our computers nowadays. We expect, when we perform a calculation, we can trust that the computer is doing whatever we've asked it to do. With whatever, if there is a mistake, is in our code, not in the computer itself. And that is possible, or became possible in classical computing only after error correction was invented and developed. And the same goes for quantum computing. If we want to have fault-tolerant quantum computing, we need error correction. And error correction usually works by basically encoding the information in a redundant way, so that we can identify errors and then correct for them. But in the quantum case, it's much more complex than doing it in the classical one, and it's really a very big challenge. So that means that that implies that fault-tolerant quantum computers are expected to be a possibility in 10 to 30 years, so it will take still a while. I myself, I'm more on the optimistic side. I think more about 10 years than 30, but it depends a lot on who you ask. What's important is that when the computers were conceived, a lot of efforts were devoted to understanding what could we do with these computers. And lots of algorithms exist, but of course they all rely upon the assumption that they are fault tolerant. Nowadays, we are in this other era, which we call the NISQ era, noisy intermediate scale quantum computers. They are noisy, meaning that they are prone to errors. And they're unavoidable right now. We have to deal with them. And they also are relatively small, that's why intermediate scale. So they have the order of 10 to 100 qubits at the moment, and they're increasing very quickly, so we will have thousands of qubits soon, but still with errors. And for that, of course, the problem is that these computers cannot use those algorithms that were conceived under the assumption of fault tolerant. But that doesn't mean that they're useless, actually, because basically these computers can be prepared, can be put into states, into quantum states, that we cannot actually simulate classically. That's how they proved non-useful quantum advantage. But that can be a very valuable resource. And perhaps not for every possible application for which one could use a fault-tolerant quantum computer, but it is for sure very useful for simulation. So we go back to the basics of quantum computing, which is simulating all the quantum systems. And this is usually done by the means of what's called hybrid algorithms. And the principle is essentially that one does as much as one can on classical computers and only uses the quantum computer for the thing that is really, really hard, which is, for example, encoding a quantum state. So we are Algorithmiq are mostly working on this framework. We're devoting most of our research efforts at the moment in the development of this kind of algorithms for NISQ devices for the specific purpose of, so it's very focused research on the specific purpose of simulating quantum chemistry. And we do it in a very, let's say, peculiar way. So we do it using something that is less common, let's say, which is using informationally complete measurements. So in quantum mechanics, when we perform a measurement, like the ones we were seeing before in Tony's talk, very often one assumes the measurement to be so-called projective. And that, I'm not going to go into details, but basically that implies that this class of measurements, the projective measurements, do not give you all the information on the quantum state. Never mind how many times you repeat the same measurement, you only see part of the state, part of the information. Maybe very accurate if you repeat it enough, but not the full picture. Instead, we use something called informationally complete measurements that actually give you the full picture, provided enough data. And this is something that we started to think about already some years ago, and we started to try on real hardware. Could we use these informationally complete measurements? Could it be useful, for example, for things that were very abstract, academic, like entanglement detection and things of the like? But then at some point the question arose, like, can we use this for something useful, like, for example, speeding up simulations in quantum chemistry? And then later on in a collaboration with IBM, we actually developed a method, an algorithm, that is aimed at speeding up the quantum chemistry simulations through these informationally complete measurements that are actually, what we do is we adapt them. So they measurements that the algorithm learns how to measure for example, the energy of the molecule that we're simulating along the path. And this leads to this very nice convergence curves this is the error of the energy along as a function of the number of times that one uses the quantum computer. It could be time, if you like, the time of the computation. And as compared to other methods, you can see my mouse, you can see that usually this just decreases as 1 over the square root of the number of shots. It's just statistical inference. While with the adaptive measurement, what we have is that we reach this point, which is chemical accuracy, the accurate enough measurements or inference in a much shorter time. And actually, this actually improves the scaling itself of the simulations, of the calculations, but nearly quadratic. So from something that scales like n to the 6 to something that scales like n to the 3. So that, of course, is a huge improvement. And this means that if we want to do some quantum chemistry simulations using some of these techniques that are quite slow, it will become infeasible, while it can be possible with this. But what's really important is actually that because we have this informationally complete data, it's not only the inference of the energy that we have. This informationally complete data can then be used to do all sorts of other things, like mitigating the noise, or more efficiently also finding how to actually prepare the state on the quantum computer, a number of applications that we are developing. So in short, what we're doing now is we're putting together all these techniques into a platform, a software platform, that can sit on top of essentially any hardware provider, because it doesn't really depend on the hardware we're using, it just relies on basic quantum information science. And it's a platform that the idea is basically that given a molecule, it tries to simulate as much as it can from using classical compute, because after all, computational chemists, very smart computational chemists, have been working for many years developing methods that work very well and can solve the problems in many cases, but sometimes they can't. And that's where the quantum computing can basically substitute that part of the calculation that the classical method cannot do with this same quantum boosted core or piece of compute, and then give back the whatever physical chemical properties we're interested in. And the fact that we're using this informationally complete data makes the communication between the classical and the quantum parts very natural and very easy to implement. So now going back to the question, and what can we do with this chemistry simulation? How can this help somehow? How accurate quantum chemistry can help in real scenarios? Then what we're considering is something along this line. So basically, once one has identified the possible set of targets, and maybe even at a polypharmacological level, because as was said before, drugs don't really, they're dirty, they don't really interact with just one single protein. Once a set of targets are identified, then we can now computationally, in this reductionist but quantum powered way, we can try to find the molecules that bind to those targets. Of course, this requires having some 3D structures for those proteins, from crystallography, AlphaFold, in whatever means that we can have. By the way, incidentally, there are also quantum algorithms for protein folding, but of course rely on good enough hardware. They're not yet useful, but they could be, and they fold from first principles. It's not a learned thing. And once one has these pockets, once one has these pockets, the structure of the pockets, one can then run the simulation to basically assess whether there is binding or not, like basically predict the binding affinity from first principles. Now, this is particularly interesting, I think, for for molecules about which we don't know anything, because there's no data about them. We cannot really do anything data-driven. And incidentally, that's most of the proteins, most of the molecules. So the space of drug-like molecules, the chemical compound space, is estimated to have at least 10 to the 60 molecules. That's a really huge space. And that means actually that the amount of molecules that we consider that right now is of the order of millions, tens of millions, perhaps a bit more, but it's certainly not even close to a very tiny fraction of the whole set of compound drug-like molecules. And one thing that quantum computing can also do, and there's been some proposals for this as well, is algorithms to explore, so basically given the pocket, to explore, not just to screen a molecule or set of molecules, but actually to explore the space to find one that could bind to that pocket very efficiently. So this, of course, would be very interesting if one generalizes it, for example, to the case of having multiple pockets that one wants to take into account, or even with negative bindings in a way that, in the sense that one might want to minimize the binding affinity to some proteins that one knows that you shouldn't perturb, right? So this, I think, summarizes a possible way forward to combine these things. I don't think it's the only way to combine quantum computing with network medicine or complement network medicine through quantum computing. For example, once we have the fault-tolerant computer and we can address even much more complex simulations, one could even think about helping with the problem of the lack of data in the interaction, because perhaps we could model these very complex interactions, not in the near term, but in the longer term. So I think there is a lot of room for improvement. So just the takeaway messages of everything I've said, I think, can be put in this way. So basically, quantum computing in general is necessary to simulate some of the chemistry that plays a role in biological processes. That understanding human biology is something that requires a holistic approach, and network medicine is the way. That quantum computing and network medicine are both becoming a reality, so this is just happening now. And the combination of these two can be really transformative for a industry that basically seems to be failing, precisely as Professor Loscalzo said, because of this reductionist approach in which everything goes very well in vitro. The molecule binds to the protein until you try it on a living being, and then it turns out that it interacts with everything else, and it just doesn't work. So that's all. Thank you very much for your attention.
Okay, thank you, Guillermo. And now we hope that there will be also some discussions. And yes, Professor Loscalzo. Again, this is adapted to my loud voice. So.
That's fine. Thank you. That was a terrific summary. I have a few comments that we probably could discuss through the afternoon as well, that just take off on your last couple slides. I mean, the first is that the instances of protein structures that are used as the basis for in silico modeling are static images, right? So they're the best crystal structure that we have, or they're the best NMR structure we have. But as you know, proteins breathe, they change shape.
Yes.
If you look at the structures, the imputed 3D structures from cryo-EM, there are hundreds of them for a single molecule. So if there were a way by which one could begin to think about the dynamic changes in conformation using quantum approaches, that would be a terrific sort of a distribution of possible combinations from which you could discern which molecules might be the best to drug a protein. That's the first point. Second point is most of the simulations, including the ones that my colleague Felice uses and the pharmaceutical industry in general, they'll wind up looking at one or maybe two pockets. And the pockets they identify are purely geometrically structurally defined. Right? Is it big enough to fit this molecule in? And then we'll work from there. But for the very same reason, in some cases, the proteins become conformers. So you add a molecule to it, and now the protein can adapt and change its shape and facilitate the evolution of a new binding site. Never taken into consideration for most of these issues. The third is, in the area that's dear to my heart, can you drug a protein-protein interface? So medicinal chemists will say, no, you can't do that. Until recently, they would never look at such a molecule because they would simplistically say that that interface is largely a flat surface, and unless you have a planar organic molecule, you'll never get it in there. But that's, of course, incorrect, that interface has peaks and valleys and edges and geometric edges and pockets, just as the exofacial site does. So ways by which to define the interfacial sites and the novel binding sites that might be created as a consequence of two proteins binding together is another area that requires, I think, enhanced computation. I can go on. Let me ask one. I'll ask one more. Make one more comment, and then we can carry this on afterwards. The last point is allostery. Right. So allostery means when can you change the shape of a potential binding site for another macromolecule as a result of binding a second, usually small molecule to the same protein. In fact, we in network medicine, looking at protein interaction networks, assume that each of the binding interactions between a protein and its other 10 binding partners are independent of each other. That's absurd. Right? That's absurd. It's a complicated problem. How can the binding of one protein modify the binding of others? And what ultimately can wind up in the complex? All 10 proteins on average, or 3? Are there some restrictions? Is there competition? So that's yet another complicated computational problem that I think would benefit from, potentially benefit from quantum approaches. So I've talked too much.
Thank you very much. Yeah, actually, regarding the first point, this is something that we've also been thinking about. Of course, this is still working. Yeah, okay. It's also something that at some point we were also discussing how to actually incorporate, because of course the change of conformation of the molecule after the binding, it's also it's an important aspect. I think it's, of course, we have to go one step at a time, but that would be very interesting to, for example, incorporate some sort of molecular mechanic calculations, like a kind of, if you like chopping the calculation into different parts. Because the electron correlation is not relevant everywhere at the same time. It can be almost everywhere be disregarded. But there are parts in which it might be relevant, like for example in the binding itself, in the binding pocket itself in some cases, or but maybe one could complement this from a contribution regarding the change of shape that could be modeled in a very rudimentary way using something like molecular mechanics. That could be maybe something interesting to look at. And then when it comes to the binding, I had a question back regarding one of the points that you asked, which is, so you were considering something would go more in the direction of, rather than perturbing the network at the level of knocking out a specific protein, more like controlling the actual links, right? So perturbing at the level of the links, or if I understood correctly.
Yes, because I think the all or none, the knockout experiment is not generally relevant to most of what we do from a therapeutic perspective. So fine-tuning interactions, or maybe even allowing for the binding of another binding partner that's inhibitory, would probably be, or could be, equally therapeutically useful.
Okay, I see. Thank you.
So let's see if there are more questions or comments. Yes, Eric.
So thanks for a very enlightening talk and a very clear presentation. What's your view on quantum biology? And by that I mean real phenomena of entanglement and intrinsic quantum properties in biological systems, which typically take place at room temperature and in water solution.
Well, I have to say that I don't think I'm really qualified to give a proper answer to that question. I think I've heard that, or I know of several examples that people believe in which people believe that quantum mechanics play a role, like the sense of smell and some of the cases. What it is clear, I think, from this is more closer to my expertise, is that in some processes, like for example, quantum transport in some networked systems, like the FMO and so on. In that case, we've worked on that in the past, also with Matteo and Sabrina. It seems clear that in those cases, quantumness plays a role, but not only. And the interesting thing is that, of course, we usually don't assume that this should be the case, because quantumness is always associated with this complete isolation and purity of the system, while, of course, in biological environments, this is not the case at all. But in this case, for example, it's interesting that this is a biological system in which it's clear that the function of the system itself is enhanced by the noise. So you need randomness, you need quantumness, but you need as well noise, quantum noise. So in some cases, I think it's, I would say it's quite clear that there is some randomness, but I'm not sure about human biology, of course.
I can add maybe a comment and further clarification in the direction of what Guia just said. The first comment is related to the fact that in many of the examples, you see, when we had this multi-scale approach that Guia was presenting here, and we were representing some of the network medicine networks, we are not assuming that they are quantum. I just want to clarify these things. Those networks are, we are assuming that those are classical networks, and then we can complement with quantum tools computational problems of network medicine. So this is just a clarification. On the contrary, in the case of quantum biology, and this is also something we are interested for sure, but it's a different framework in which there is some sort of proof, even experimental, and there is a lot of activity of networks in biological systems which are actually supposed to be quantum in the sense that they are described at some quantum transport properties. So I just wanted to make this distinction. In that case, of course, as you pointed out, these biological organisms, they are living in conditions which are certainly not isolated, so they are very noisy, and they are not the standard conditions of of quantum information processing. We just, from the talk of Tony, we heard that generally noise is something which is destructive. So the environment is something which destroys quantum properties. However, again, this is in my background. I've been studying actually noise in quantum systems for all of my career. This idea that noise is necessarily, an enemy for quantum systems, especially when it is applied to complex systems, it's not so straightforward. So what can happen actually is that a suitable interplay between coherence and the action of noise could be the factor that enhances transport. And this is what is believed, for example, in the FMO complex, in quantum biology, and so on. So it is not necessarily true that noise, because of the presence of noise, we won't be able to observe any quantum enhancement in this case, but actually perhaps it is the interplays between these two things. And it is a very difficult problem to model, so therefore it's difficult to gain a proper understanding, but this is what I believe the state of the art of in quantum biology. So unfortunately Martin is not here, but I think that I believe this is also his perspective on this. So more questions? From the audience? Okay. From the audience, meaning from the online audience. We will have soon to wrap up, but we will have time for discussing with some of you further these discussions. And of course, for us, this is just the starting point. I would like then to summarize. Guillermo, maybe you can leave the presentation, because I would like to summarize briefly what we tried to achieve during this symposium. And I think for me, what is really important is to have an occasion to start to spark really a discussion and to spark potential collaborations and to at least what we really truly believe, try to find what is important for also for quantum computing and quantum algorithms, that is interesting applications and use cases. So on the one hand, I think that quantum can provide many tools for complementing network medicine. On the other hand, the network medicine can provide very useful use cases for quantum computers, because we are at the very early stage of quantum computing and quantum simulations, and even understanding what are the topics, what are the applications for which this discipline can really contribute is crucial. And so we believe that this is a tremendous opportunity for these fields to be together, and therefore we hope that we will continue these discussions in the future. And I would like to thank everyone, both our speakers, first of all our speakers, but also all the audience and all the online participating people. And thanks a lot. This is just the beginning. I'm sure that a lot will follow up from this. Thank you very much. And of course, thanks to Guillermo too. He's the only one I didn't thank.